आने स्टार्ट होते हैं तो आ जाए ना, क्योंकि वो आ जाएंगे ऑस्ट्रेलिया वाले। मुझे लगा था दोनों ना उनके टाइम लेट है ना, दे माइट कम 2:30 के साथ। मुझे उम्मीद था वो कोई दिक्कत नहीं।
We started off with some basic lectures, starting with Thymus and catching up on with some basic, components, innate immune components. Then we talked about complement. It was very nice talk on complement. Then regulatory mechanisms in innate immune response. Then we had a very good lecture on MHC-linked control of immune response. Then pattern recognition receptors and inflammation. And finally, in the evening, we had two great lectures, one by Michel Pallai on approaches to human immunology, and then on B-cell responses by Joshi Jacob. So everything was going great, is going great. And today again, we have a very exciting program. And I welcome you all to the day 2 program. And I have the pleasure to introduce the moderator for today. We start with session 5, and the moderator is Professor Sarath Dalai. Let me introduce Professor Sarath Dalai. He is Professor of Biotechnology, as well as Director of the Institute of Science at Nirma University in Ahmedabad, India. He's basically physics graduate, but later on did masters and PhD in biotechnology from Jawaharlal Nehru University, New Delhi, which is a very prestigious university in India. Then he had postdoctoral training at a couple of universities, very big institutes in US. One is NIH. And then he moved on to Johns Hopkins School of Medicine in Baltimore, USA, before he came back to India to join Nirma University. He has a very strong 20 years teaching immunology to graduates and postgraduates in the university. His research interests are the training in malaria vaccine development program and addressing the underlying issues related to maintenance of memory CD8 cells vis-a-vis sterile protection induced by whole sporozoite vaccine. So I hand over the session to Professor Sarath Dalai. You have two speakers, so Sarath, the stage is.
Good morning. Thank you, Professor Arora, for those kind remarks. I'm sure all these participants and panelists have enjoyed the lectures yesterday. Today also we'll have very exciting lecture. I'm going to moderate these two sessions. One is Dr. Jana Groom, another is FIMSA President Laura Mackey. And Dr. Groom is going to talk about T-cell activation and differentiation. I'm sure all the students are very keen to learn about T-cell activation and differentiation. We all love T-cells, although B-cells also we love, particularly at the time of Corona. So, in fact, Dr. Groom's, his research focuses on how immune cell location and interaction control the immune response. In fact, she developed her interest in immunology, particularly in the B cells, and during her PhD, and her research revealed a novel mechanism of autoantibody production, which was pivotal in licensing BAP blocking antibody for lupus treatment. During her postdoctoral training at Massachusetts General Hospital and Harvard Medical School, she found how chemokine regulation was not only critical for T-cell positioning, but also important for T-cell priming. She is now head of the Immunology Division at Walter and Eliza Hall Institute, and she uses advanced in vivo and 3D imaging platforms and molecular analysis to dissect T-cell interactions that independently balance fate decisions between T-cell effector function and memory formation. And in fact, this is something we would like to learn, greatly, in coming time, because formation of memory T cells is still, to many of us, an area which is evolving, and we should learn more and more about it. Our goal is to identify context-dependent strategies to harness this T cell differentiation axis, either to stimulate T cell effector function for the elimination of chronic infection and cancer, or to amplify the formation and longevity of cellular memory to prevent current and emerging infectious diseases. So I'm sure today she is going to talk about both CD8 and CD4 T cell differentiation. I'm sure she's also going to brief us the latest research she's doing with respect to SARS-CoV-2 infection. With this, I would request Dr. Bloom to deliver her talk.
Thank you, Sarah. It's lovely to be here with you this morning, or this evening as it is in Melbourne. I will just share my screen. And hopefully if I start presentation mode, you will let me know if it's working for you. How does that look for everyone? Good?
Yeah, it looks good.
Yeah.
Excellent. I'm very happy to hear that everyone is excited by T cells. As I was introduced, I'm really interested in the biology of cellular interactions, and so that means the way T cells interact with not only dendritic cells during priming, but also B cells during a germinal center reaction. And so I think this is a really nice topic to discuss today. I won't go into too much work about SARS-CoV-2 research, except to give a really primer, because I saw on Sunil's list of presentations coming up that there are a few talks in this area. So I didn't want to stump some people who are talking after me, but yeah, so as I said, I'm a lab head at the WEHI here in Melbourne. This is what the WEHI looks like, except now it's actually, we've been in stage 4 lockdown for so long, I can't actually remember the last time I saw the front of this building, but I'm sure it most certainly does look the same as I remember it. So I thought today I wanted to just give you an outline of what I'm going to talk to you about. And I know that you had a lot of discussion yesterday about the innate immune system. And so I really wanted to give you, set the scene for the adaptive immune system, because I feel that it all starts from this really intense cellular communication between dendritic cells and T cells. And one aspect that we don't often talk about that I think is critical to this is understanding cell motility and location, and how this might influence the different outcomes of T cell priming. I'll then introduce dendritic cells, and these are really the bridge between the innate system and the adaptive immune systems, and the 3 signals required to prime both CD8 and CD4 T cells. And then to give you just a quick glimpse of why this is important, I guess, and how we can manipulate this to treat infectious disease, but also to develop strategies that really make our T cells fit for purpose, I guess. So firstly, just to give you an overview of the adaptive immune system, this is when a pathogen, in this case demonstrated by a virus, infects cells. And here we see infecting the epithelium. And this is detected at the first point by dendritic cells. This process then activates these cells, and this leads them to be able to move into the secondary lymphoid organs, in this case, the lymph node, but also the spleen. And here, this is where we get our really close interactions between dendritic cells and T cells, and also B cells. And this leads to T cell, the adaptive immune cell activation, and a development of their effector function. Following this, these cells can then move back into this area of inflammation to really, either in the case of antibodies, as you heard yesterday, neutralize the virus, or to eradicate the virus and viral infected cells. So this process takes time. It actually takes a fair few days to a week. So given that the innate immune system is really rapid, why is it that we would bother with the adaptive immune system? It takes a few days for T and B cells to be stimulated in secondary lymphoid organs, and then for their expansion and development of their effector functions in order to remove the infectious agent. And so why would we bother with this secondary system? And the answer really comes down to this graph. And this is on the axis here, we have either antigen-specific immune cell numbers or the concentration of antibody. And before an infection, the levels of these is very low, and these rapidly increase during an immune response. And when this is finished and the pathogen is eradicated, we're not left with we don't return to the naive state. We have this increase here, this reservoir of memory, and this is really the critical reason why the adaptive immune response is so powerful, and why it allows us to manipulate, leads to manipulations such as the formation of development of vaccines, because having this repertoire of memory cells allows, if we ever encounter the same pathogen again, a larger increase in the response, which is more rapid. And so really the time taken for this response is really worthwhile, because it means we have absolute protection from disease from following encounters. So where does this take place? Well, I know that you spoke a bit yesterday about the thymus, and I don't want to go over the maturation of T cells in the thymus, except to say, for me, this is where the adaptive immune response really starts, the system is established, because this is where our mature but still naive T cells, CD4 and CD8 cells, are generated. Each of them express either a CD8 receptor or a CD4 receptor, but not both. And importantly, these T cells don't recognize free antigen in its natural conformation. They recognize antigen via their T cell receptors in the context of antigen presentation. For each T cell in the thymus expresses a unique T cell receptor, and this means that there are millions of different antigens that can be recognized. Once made, a CD4 and CD8 cells need to leave the thymus. So where do they go and how do they get there is the next question we will ask, except I also wanted to give some Australian history and let you know that the function of the thymus was discovered by Jacques Miller. And if I was still going into a WeHi on a regular basis, Jacques and I have offices next to each other. So our T cells leave the thymus, and they do this through the process of migration. And this migration is really, from my perspective, the feature that sets our immune system apart from other cellular systems in our body. Without the ability to migrate and to interact with other cells, none of what I'm about to tell you would be possible. So it's really important to consider this when you look at schemes of T cell differentiation, that you remember that these are really dynamic events. And so here we have our wandering, migrating, naive T cell. And these T cells enter the lymph nodes due to their expression of a chemokine receptor known as CCR7 via high endothelial venules. They then scan around the T cell paracortex in search for a dendritic cell that may be expressing its cognate antigen. If they don't see such a dendritic cell, then they leave into a downstream lymph node and repeat this scanning behavior until they do indeed come across an antigen of interest. For the dendritic cell part, they also enter lymph nodes through the afferent links, again due to the expression of CCR7, and this allows them to translocate through the subcapsular sinus and enter into the paracortex. And this location is where T cell priming occurs. So I'm hoping that this movie can is working well for you. And I just wanted to show you how dynamic the movement of scanning T cells are. You can see them here in green, whizzing around the paracortex of a lymph node in search of a dendritic cell that may be presenting its antigen. They're very quick as they pass over dendritic cells, looking to see if they have an antigen of interest. But it's also more than migration that's important, it's also the location. And so secondary lymphoid organs such as lymph nodes and the spleen are really highly organized. And this allows T cells to come into contact, into conversation with different cellular interactions and different T cell priming. So I'm going to focus on the lymph node here, but the same context really also applies for the spleen. So you can see here there are several different regions within the lymph node. The B cell follicles and paracortex, of course, and the subcapsular sinus between these B cell follicles. And these, all of these areas really host different cells that can influence T cell priming in different ways. And where and how T cells move to different areas can then therefore really determine the type of effector cell or memory cell that they become. And this is also dependent on the type of antigen or the context of infection, whether it's a viral infection, or a helminth, or a fungal infection. And it's important to remember that this is not, that these are three-dimensional structures. So when we look at things like histology, it's important to really understand whereabouts you are in the organ to give you a true knowledge of the location of immune cells. So as I mentioned, T cells cannot recognize free antigen. Their protein antigens must, long protein antigens must be degraded into individual peptides that range from about 8 to 15 amino acids long. And this requires the process of proteolysis. And this occurs in an antigen presenting cell. There are different types of antigen presenting cells. Dendritic cells are the best type of antigen presenting cell, and they've been given the name of professional antigen presenting cells. And we also have macrophages and B cells that are also capable of presenting antigen. And what you can see here is that each of these cells has a preference for the type of antigen that they prefer to present. And this is one feature that gives the immune system flexibility to really respond to diverse infections. Moreover, as we know, immunologists love to subset subsets. And so dendritic cell, within the dendritic cell lineage, there's actually multiple subsets. And again, these have very specialized functions. Here we see that the CDC1 population present primarily to CD8 T cells. CD2s present mainly to both CD4 and CD8 cells. And monocyte-derived dendritic cells don't directly don't directly prime T cells themselves, but their presence can influence the way a T cell differentiates. So again, we see that the different cells that a T cell comes into contact with can really tune and shift the immune response. So this is an image of dendritic cells, the dendritic cell network, and here we see it in the spleen. And dendritic cells really line our barrier surfaces here in the skin, but also the mucosa and lung. And this helps them in their role for the detection of pathogens. They are the ones in our immune system that really sounds the alert to infections. So what does it take for a dendritic cell to go from the skin, say, to presenting antigen in a lymph node? Well, upon recognizing a pathogen and the pathogen-associated molecules, these dendritic cells start to activate. And this process allows them to process antigen proteins and display them in the context of MHC, major histocompatibility molecules. So here we can see a cross section of our APC down the bottom and our T cells up the top. And here we have MHC class 1 pathway, and this pathway presents to CD8 T cells. MHC class 1 is expressed on nearly all cells of the body, and it presents peptides from endogenous proteins. And these proteins are loaded onto the MHC class 1 in the lumen of the endoplasmic reticulum and then presented on the cell surface. In contrast, MHC class 2 presented specifically to CD4 T cells. Now, MHC class 2 molecules are expressed only by antigen presenting cells, and these present peptides that are derived from exogenous proteins. And so these peptides are generated through the endosomal and lysosomal network to move onto the cell surface. And another bit of Australian trivia in this pathway is that Peter Doherty made the discovery that T cells recognize self-MHC molecules that have bound pathogenic antigens. And this discovery was awarded the Nobel Prize in 1996. So back to our dendritic cell. We now have antigen being presented on the cell surface, and this activation process has also led to the upregulation of our chemokine receptor CCR7. Again, this allows these cells to move into lymph nodes and transmigrate into the T cell paracortex. They also upregulate co-stimulatory molecules, and here all of these features really allow them to present to a naive T cell in the lymph node. So again, to reflect on how dynamic these processes are. When we first look at a T cell coming into contact with antigen presenting dendritic cells, we see short scanning movements over these dendritic cells. Probably a little longer than if peptide wasn't present, but still relatively short. And then later, a day or so later, these contacts really move. Here we can see the antigen specific T cells in green, really having long-lived contact with antigen presenting dendritic cells. And so essentially, this is the process where dendritic cells pass on the information to T cells that they've seen an infection out in the periphery. So what does this information, what is the details given in this conversation? Well, it really comes down to these three signals. From up the top now we have our antigen presenting cell, and down the bottom we have our T cell. So signal 1 is the presentation of the specific antigen in the context of MHC, and this leads to the activation of this T cell. However, this signal on its own isn't sufficient to lead to the effector function of this cell. In fact, with this signal alone, the T cell interprets this as a tolerogenic response, that they may be seeing a self-antigen. And that's why it's important that it's done alongside co-stimulatory molecules. And there are a few of these, but here we see the classical CD28 and CD8. And thirdly, we have cytokines, and these lead to really the differentiation process. And here again, I'm just showing one cytokine, but there are multiple different cytokines that can really tune the way a T cell responds to a particular infection. And this really allows, it tells this T cell the kind of effector cell that is needed for a particular infection. And so this is more the common way of looking at this, where we have our dendritic cell and our T cell and our 3 signals being given. And again, here we see type 1 interferons and IL-12 being given to the T cell. But again, this can be other cytokines, as we'll go through. Also, this interaction also really leads to a feed-forward loop, where our T cell can also give cytokines back to the dendritic cell. And this can lead to further activation of those dendritic cells, but it can also lead to their death. And this is thought to be a process to dampen the immune responses. It's important to remember that this interaction occurs in the context of a polyclonal response. So while this T cell is recognizing its specific antigen, there are, again, millions of other T cells around repeating this scavenger behavior that I told you about. And these may come into contact with some of these cytokines, but because they're not getting signal 1 and signal 2 from this interaction, they continue to be naive. And it is just this one antigen specific T cell that is clonally expanded and differentiated. Again, these are really dynamic interactions, and one dendritic cell can either sequentially interact with T cells or at the same time interact with 2 different T cells, as we see here. And this can either be multiples of CD4 or CD8 cells, or it can be either of these cells. And when this occurs, these cells can then both influence each other's differentiation in addition to the dendritic cell. So just to look very quickly at T cell, CD8 T cell differentiation, we have our 3 signals here. And this really, for T cells, leads to a relatively easy decision. Based on the cytokines and the type of dendritic cell, these can either become cytotoxic effector lymphocytes, and they express huge amounts of interferon gamma, granzyme B, and perforin. And this makes them really exceptional killers of viral infected cells and also tumor cells. The alternative is to become a memory precursor, and these cells express a lot of IL-2, and these are the ones that will remain after the pathogen is cleared, and will lead to long-lasting protection and more rapid secondary responses following the re-encounter of the same pathogen. So here I've shown this really as just one single memory cell, but more and more information is arising every day about the different types of memory and the different features that all of them have, and how they work together to really establish this long-lived memory. And so I didn't want to go into that too much because I know that Laura will discuss that next. But just to say, this is, again, a very simplified scheme, and that within these there are still multiple cell types. But for CD4 T cells, the scene is a little bit more complicated. We have multiple effector lineages, and these are known as helper cells because they help other cells of the immune system respond to a specific type of pathogen. The differentiation of each of these Th1, Th2, Th17, T follicular helper cells and induced T regulatory cells is determined by that third signal. And we can see here examples of these cytokines that really skew the differentiation down a particular pathway. However, while they're shown here as really distinct responses, in reality, whenever there is a pathogen encountered, we likely get some mix between these effector subsets. But one of these will predominate that is really fit for purpose. So for instance, Th1 cells are very good at eradicating viruses. And so this is the other side of the differentiation. These cells are really, I did want to say just quickly that really this differentiation program is really established by a key set of lineage defining transcription factors. And these really enforce the differentiation of a particular lineage. And in addition, as I'll come back to, they also block the counter differentiation to any of the other lineages. And so this is more looking at the effector function of these T helper cells. They're really characterized by the cytokines they express, and this has a direct effect on other effector cells in the immune system, as I said. So Th1 cells express interferon gamma. This leads to activation of macrophages and neutrophils and epithelia, and this really helps the lysis of infected cells. For Th2 cells, they express IL-4, 5, and 13, and really target basophils, eosinophils, and epithelia to assist with worm expulsion. For Th17 cells, they express a range of IL-17 molecules, and these act on fibroblasts and epithelia, and this is essential for the eradication of extracellular bacteria and fungi. And these together, Th1, Th2, and Th17, really are, I guess, the basis of the T helper effector cells. And so I like to group them together and think of T follicular helper cells and regulatory cells a little bit separately to this, because while these cells act against a specific type of infection, really these cells are differentiated alongside these other effectors, and they can be seen to arise in most infection types. And so coming back to this idea that this is really a tight balancing act between these differentiation fates, different factors from the cells presenting from the cytokines and microenvironments within lymph nodes or spleen really influence what this cell, the fate of this cell, and how it will differentiate. And this is also influenced by other factors such as the affinity for the antigen. And here I'm showing just this balancing act between TFH cells, T follicular helper cells, sorry, and Th1 cells, and how where we have high levels of other, it usually leads to a lessening of the reciprocal population. And this is true not just between TFH and Th1 cells, but also true of that counter-regulation between all of the helper effector cells that I've mentioned. And so understanding this balance is really important for how the immune system functions to efficiently respond to a pathogen. Now, multiple studies that I'm sure you will hear about during the course have now really quite clearly shown that there is a disrupted balance in patients with severe COVID-19. And so this is really critical to understand exactly what are the mechanisms at play that are steering the immune response in an unproductive way. So finally, I just wanted to leave you with this thought that really understanding these pathways for T-cell priming and differentiation is really critical, not only for us understanding how we respond to a pathogen and how we can best overcome it, but really important to identify new ways to make our T-cells fit for purpose. And so here we're looking at a vaccination protocol where we can elicit both CD8 and CD4 cells. And this can be done in the context of either fighting an infection or eradicating a tumor cell. And what we want from each of these is really quite different. If we want a vaccination to provide long-lived memory and efficient humoral responses, then we really need to drive our T-cell differentiation towards our memory responses and towards our T follicular helper cell differentiation. However, if we want a vaccine to overcome an immediate threat, such as the presence of a tumor cell, then the promotion of CD4 and CD8 effectors is really the desired outcome. And so really understanding the processes of priming, T-cell priming and differentiation is really essential to generate these new line of vaccines. And so that just leaves me to thank you for your time and thank you for the invitation and to open up for questions.
Thank you, Dr. Krum. We have several questions. Let us see whether time is going to permit. First question, are the memory T cells strictly specific for the epitope which generated them? If so, then is the probability of the same epitope reaching the memory T cell high or low? And in case memory T cells are redundant, then won't they be activated by multiple different epitopes? Although evidences are present where single amino acid substitution can change the response type from Th1 to Th2. So the question is that whether it is redundant. Yeah.
So yes, memory cells do have a specific epitope recognition, and so that's really critical. That's exactly the way that memory works. That we have a response if we encounter the same pathogen. And so that is critical. That's not to say that it necessarily needs to be the same infection. Some of these epitopes that a T cell might see can overlap with very related strains. And I think there are a few examples of that in COVID-19 and SARS-CoV-2, where we see some people have memory cells from other coronaviruses. But the actual epitope that's recognized is conserved. That's a very important point when considering memory. The second part of the question is really interesting. So just because a T cell really predominated a Th1 type response to start, it doesn't necessarily mean that upon re-encounter of that infection, it will go in exactly the same way. It seems there is some level of epigenetic regulation to lead it down that same path, but it's not necessarily so. And between the helper cells, there is a fair bit of plasticity, and cells can move from one helper subset to another, even during the course of an infection.
But we do know that Th1 cells are more plastic compared to Th2 cells. Am I right?
Yeah.
Second question, I think similar, but let's go to the third question. All nucleated cells express MSC1 and present process antigenic peptide to cytotoxic T cells. Mainly dendritic cells, macrophages, and B cells express MSC2 and present process antigen peptide to T helper cells. Can we say almost all cells of the body are antigen presenting cells? As you use them, term professional cells, antigen presenting cells, can we assume all cells which can express MSC2 are professional antigen presenting cells? I'm sure some MSc students have asked this question, but definitely valid question. Yeah.
It's a very valid question. And I love the notion that every cell in our body is an immune cell. I would say that every cell can present antigen, but I still wouldn't say that they are professional antigen presenting cells. The dendritic cells are by far superior in this process. So, yeah.
So what does detect the duration of memory? For example, it could be 5 years for one disease, lifelong for another. Let's say measles, it could induce lifelong, or chickenpox, maybe 12, 15 years. Malaria, maybe 1 year, less than that. So this is the question. So
this is a multi-million-dollar question, actually. This is, we really don't yet understand, and Laura may have an opinion on this, but I think we don't yet understand exactly what it is that makes a T cell long-lived. We know that, some flu vaccination actually only protects sometimes for less than the flu season. So we really need to get a better understanding of what it is that's making our T cell memory long-lived. It varies a lot from pathogen to pathogen. And all I can say is it's a very good question.
Yeah, this is the question all the time we ask without getting much answer. So very nice. How TSCM, which is stem cell like memory, get differentiated from naive cells without priming?
Differentiated from naive cells without priming. I'm not aware of that paper. I've done some work on stem-like memory, and we see that there is T cell priming in the process of differentiating them, and that they can be, and that it really does occur at different sites of a lymph node to other locations. Yeah, I'm not sure of the literature behind that question. I'm sorry.
Another number-wise, which cells are more numerous among the various types of differentiated T cells, and how their numbers are regulated during differentiation?
The maximal, could you repeat the question? So how many cells are?
Which cells are more numerous among various types of differentiated T cells? Probably they're talking about T helper versus cytotoxic T cells.
Yeah, if they're talking within the helpers, which is the most predominant, it really depends on the type of infection. So if it's a type 2 skewed infection, or a helminth, or even an asthmatic response, we'll see a predominance of Th2 cell differentiation. Alternatively, if it's a virus, we see a lot of predominantly Th1 differentiation. between CD4s and CD8s, that's a good question. In our work, I find the kinetics of CD8 cells is a little faster than CD4s. But whether that is substantial enough to lead to greater proliferation, certainly from the naive repertoire, it seems like there are more precursors of a single epitope in the CD4 repertoire than the CD8 repertoire. No, actually, the other way around. There's more CD8s than CD4s. So I think combined those two features, maybe there's probably CD8s outweigh CD4s, but yeah, it's a tough one.
So what is the half-life of memory T cells? I think it's a similar question. So, but it's more specific. What is the half-life? Can we define half-life?
I don't think we can define half-life because there are so many variables, and it depends on the infection, it depends on the priming, it depends on the subset of memory that you're actually looking at, and because we know that it varies so greatly, I don't think we can comment on a half-life.
It's okay. One participant has complimented your presentation and wanted to ask question. Would you like to say something on your asymptomatic COVID-19 people? So can the vaccines work for long term? Then these asymptomatic patients are because of antibodies against COVID-19 and some coronavirus providing partial immunity. What is your thoughts on it?
I think we'll take this question in the evening because that's a specific session we have on COVID. So we need to wind up.
So if you do not mind, I have several questions, but I just want to ask one question. When we are talking about signal 1, 2, and 3, do you think it follows certain kinetics? Let's say while antigen.
No, I don't. I don't actually. I think they're more or less simultaneous. And yeah, I think that is deceptive a little bit that we call them 1, 2, and 3 as if they're occurring in that order, but they're really simultaneous signals. Yeah.
Okay. So thank you. It was very nice talk. A lot of learning for me. I'll send you questions through email. So
Thank you.
Thank you. So we'll now go to the next talk by Dr. Aramake. Hello, good evening.
Good evening.
I hope you are doing all right. So
yes, very well, thank you.
Professor Laramake is the president of FIMSA. Many of you know, yesterday also she was moderating a session. She is currently at Peter Doherty Institute at the University of Melbourne. And also she has appointment in A-Star in Singapore. She is a Howard Hughes Medical Institute and Bill and Melinda Gates International Scholar. Sylvia and Charles Bertel Charitable Foundation Senior Medical Research Fellow and an NHMRC Leadership Investigator. She is the recipient of many awards, including 2019 Prime Minister's Prize for Life Scientists of the Year, Woodward Medal in Science and Technology, Fitzgerald Prize for Human Immunology, and Victorian Young Tall Poppy Award. For a number of years, she has been at the forefront of research into tissue-resident immune cells and their role in local immunity. The focus of our laboratory is on molecular signals that govern tissue-resident memory T-cell differentiation with a view to harness these cells for the development of new immunotherapeutic strategies against the disease. In fact, this is something very close to my heart as I work in liver stage immunity. we are not yet sure the T cells which are present in the liver, we think protection is ensured because of that, where they are generated and whether it's in liver or the draining lymph nodes, whether they stay only in liver or they migrate from the air, temporarily. So this is something I would personally like to learn. I'm sure students would also like to learn, particularly in the context of this current pandemic. This local immunity is very critical. As I teach vaccinology courses, some of the things I put to students only questions without giving much answer. I'm sure they're looking forward to learning many things from your talk. Thank you, madam, and I request you to deliver the talk.
Thank you very much. And thank you for the kind introduction. Just to check whether you can see my screen.
Yes, yes.
Fantastic. So as it's quite nice just to follow on straight from Dr. Groom, who mentioned to us how there's a lot of sort of within the memory T cell pool, there's a lot of heterogeneity and there's a lot of different types of memory T cells. And so when we think about protection, what's really starting to flavor our thinking is that if you want immediate protection or the best protection against infection, you might want to target those local responses that are in the tissue to rapidly deal with an infection. And this is a kind of relatively recent notion that's only come to light over, I'd say, the past decade, that not all memory T cells are equal. And certainly there are some that just reside within the tissue. So for my first slide, I just thought maybe we should think about immune protection in the context of SARS-CoV-2. And I work on tissue resident memory T cells, but always we should try and have a bit of a holistic view of the immune response. And there are so many different components that are important. But when you think about the induction of adaptive immunity, you've got all the different flavors of CD4s, you've got CD8s that will go and kill, you've got Tregs that will suppress inflammation. Of course, you will T follicular helper cells will help B cells induce antibodies. But when you think about a vaccination, which a vaccine strategy, you really want to induce long-lived memory. You're thinking about inducing memory T cells and memory B cells that will give you long-lived pools of antibody. And the type of memory T cells that you want, now we're starting to sort of think more about these tissue-resident memory T cells. People are starting to consider the fact that this flavor of memory T cells, the way that you induce them, the way that you keep them alive, they don't play by the same rules as those in the circulation. They have specific requirements. And so understanding these requirements is quite vital in thinking how to induce tissue resident T cells. And in this case of SARS-CoV-2, you would want these cells to be within the lung. So when you think about how memory T cells are generated either by a virus or following vaccination, as we've gone through in Dr. Groom's talk, naive T cells will see their antigen on dendritic cells. In the case of a virus, you'll get a huge effector T cell pool, which can go and clear the infection, directly killing infected cells. But those that are left behind are our memory pool. And as mentioned, that is not a homogeneous population of memory T cells. What we have are three subsets: our effector memory, or TEMs, our central memory, or TCMs, and our tissue resident memory T cells. And this is actually getting really complicated now. For simplicity, I'm just talking about the three subsets, but this effector memory subset in the past year or so has really started to be broken down into flavors. And some people now will call this LLECs, or long-lived effectors, rather than bona fide memory T cells. But when we think about our circulating T cells, whether they are the long-lived effector cells I mentioned, or whether they're these two flavors of effector memory or central memory cells, these cells are always circulating in the blood. So if you're sampling blood, these are the two flavors of T cells that you're going to isolate if you're looking for memory T cell responses. And it's when you go into the tissue, these are the resident memory T cells that you'll find, and their major characteristic, if you like, is that they don't recirculate. They're embedded within the tissue, and after forming long-lived memory within that tissue, they stay there for the rest of their life. So I just thought I'd give you a little bit of history into how we're thinking of sort of the history of memory T cells, and those different flavors. And the first paper that really showed that there were at least the central and effector memory T cells was this seminal paper published in Nature by Frederica Salusto and Antonio Lanzavecchia. And in 1999, they showed that there were, in fact, two subsets of memory T cells that had distinct homing potential and effector function. And that's here is our effector and central memory T cells. When we look into the liver, the lung, the skin, or the GI tract, or indeed within the circulation, a fraction of those T cells are effector memory. Now we know, of course, a fraction are resident memory, but back in 1999, these were classed as effector memory T cells. And also those T cells that are more long-lived, don't have as good an effector function as these T cells that you see circulating permanently. These cells within the spleen and lymph nodes, they were called long-lived central memory T cells. And these two different flavors of T cells could be separated, and this is for the CD8 type of T cell, on CCR7 and CD62L, which were just mentioned by Dr. Groom. It makes sense that our central memory T cells could be identified by high levels of CCR7 and CD62L, just like naive T cells, because these T cells are found within the secondary lymphoid organs, such as the spleen and the lymph nodes. And also those effector T cells, and this was work from in 1999, people knew that they could indeed go into tissues and keep recirculating. And depending on the type of priming that you might get from infection, it was known that some T cells would be skin tropic, upregulating certain molecules, enabling to get T cells into the skin, such as CLA, cutaneous lymphoid antigen, or those cells perhaps induced by a sort of systemic or gut tropic pathogen would induce T cells to upregulate integrins such as alpha-4 beta-7. Also chemokines, of course, are important to get into the gut. You would need CCR9. These would be induced early after infection, and they would cause these effector memory T cells to go into the blood. Now, sort of a really landmark paper that started to point towards the fact that there might be another population that would just stay in the tissues came from David Mazepust and the late Leo Francois, who were in the University of Connecticut. And this was in 2001, long before actually the term resident memory was actually coined, even though Dave has been a pioneer of this field ever since. And this paper showed that effector memory T cells, as they were then called, could be found within a range of tissues. So a very simple mouse experiment here. Remember, it is indeed very difficult to sample tissues such as the lung and gut within healthy individuals from humans. So a lot of these early studies were from mice. Very simple, take a mouse, infect it with a mouse pathogen, and several weeks later, once you know the infection's been cleared, if it's an acute infection, you come back, you look with a tetramer specific to that virus. And very simply, just look at the distribution of tetramer positive cells shown within this gate, sort of this upper gate here. And you could see that there was a range of virus-specific cells as identified by tetramer at a whole range of organs. And in the title here, Dave has called this preferential localization of effector memory T cells, but he did actually postulate in the discussion that our results suggest that these memory T cells either continuously circulate through peripheral tissues or indeed permanently reside within them. And the reason why over the next sort of 8 years or so, this kind of hypothesis almost started to fall out of favor, that it was cells that kept recirculating through tissues, is because people started to elucidate the fact that on effector memory T cells, they started to downregulate those homing molecules such as alpha-4 beta-7 that you'd need to get into the tissue. So how could effector memory T cells keep getting into tissues if they're downregulating the molecules needed to get into them? And so it wasn't until 2009 that the notion that there was this distinct non-recirculating subset that was permanently within this tissue, so they didn't need to get back in, they were permanently left behind after infection. There were two studies. One was from Dave, I'll talk about that separately, but another study came from my postdoc mentor, Frank Carboni, in 2009. And again, this was a very, very simple study, just to look at the distribution of memory T cells after infection. So here, you'll take a mouse and you'll infect that mouse with a skin pathogen such as herpes simplex. You'll transfer naive HSV-specific transgenic CD8 T cells. They're congenically labeled with the marker CD45.1, so you can track these cells once you put them in. They're naive, so they need to be boosted up by the virus, and you just look for the distribution of these cells over time. Now, when you infect a mouse with herpes simplex, on the skin, on the site that you infect, you get a very characteristic zoster-form lesion here that appears on the skin. And T cells will, of course, flood in to that infected site, and that's exactly what you would expect. But if you look here, looking over time at the number of these CD8 positive T cells that come in, you can see in the left flank, which is the infected flank, a high density of cells coming in. But just flip the mouse over and look on a non-infected part of skin, you can barely see any T cells coming in there. And then if you look at these later times, and this is within weeks, so this is going out almost a year later, this is a cleared infection in the mouse. It's not latent, this herpes virus within the mouse. And you can see that there is this fraction of T cells left behind specifically at the site of infection, and specifically on that left flank and not on other sites of the mouse, showing that there's this pool of memory T cells left behind where you've been infected. And to show this more, and I should just mention that markers at this point in time were associated with these T cells that would be embedded within the skin. So if you look at the same specificity of T cell, our GPT-1 T cell, which is our transgenic T cell, those within the skin were shown to upregulate CD69 and CD103. These were later classified as sort of bona fide markers for resident memory T cells by subsequent studies over the past decade, whereas those memory cells in the spleen lack these markers. Now, this is probably the more powerful experiment to really show that these T cells were indeed resident, and this was done by a skin transplantation experiment. So you take a bit of memory skin that's resolved from this previously infected mouse, and you stitch that piece of skin onto a recipient mouse here. And you look in the graft for our GPT-1 T cells. I'm not sure if you guys can see my arrow. Is my arrow working as a pointer?
Yes, it is working.
Oh, perfect. Thank you so much. So you can see these T cells are hanging around in the graft of the skin, not in the spleen, but you can't see them. And this was a control piece of skin here that didn't come from a previously infected site. So you can see that these T cells hang around even when you transplant the skin onto a different recipient. Now, the back-to-back paper here came from David Mazepust's group, who originally many years before proposed this idea. And he did a very similar experiment following gut infection. And again, he transplanted the small intestine that would have infected sort of memory T cells against the infection into a naive mouse here. And following, and if he looks in the graft here after infection and he goes into the graft, he sees that T cells are taken across within that graft, but you don't see recirculation of those cells. So this really kind of led the dawn of, changing people's thinking of the fact that there is indeed a third population of memory T cells, those that are very long-lived and sequestered within tissues. They're indeed found at high densities at sites of previous infection or inflammation, so they're left behind after infection, and they're poised, they're ready to rapidly respond if you see that pathogen again. It's been shown that they're distinct from T cells in the circulation. Various markers have been associated with these T cells, but it's quite different between different tissues, and I'll mention that later in the talk. And it's been shown over the past decade now that it's these T cells that are on the front line that have a real kind of heavy lifting role against and providing local immunity against infection, also cancer, and also having a role in mediating tissue pathology. And it's been insinuated that these resident cells are also associated with various autoimmune conditions. And this is just showing some associations that are out there in the literature. It's been shown that after infection, TRM cells have been shown to be in the tonsils after EBV. They've been shown to hang around in humans, and those that are, say, influenza specific have been shown to hang around in the lung. T cells specific for hepatitis have been shown to hang around in the liver, and so forth. In skin conditions, it's been shown that very local sort of pathologies that keep coming back at the same place at the same time, resident cells shown by that specific and distinct phenotype, have been shown to be associated with conditions such as alopecia, vitiligo, and psoriasis. Of course, CD4s are important in these conditions as well, but it's shown that the CD8s in there are of the tissue resident flavor. And also it's been shown in several human studies now that tissue resident cells can be carried across within transplants. So if you transplant an organ, these tissue resident cells can be transplanted across. And I'll mention a little bit later that these cells with a tissue resident like phenotype are now being found in various cancers where they are associated with a better prognosis against cancer. So I just thought I would sort of just articulate some of the ways where you would really define a resident cell. There are indeed certain markers, but this is getting a bit murky as you look between different tissues. But one of the sort of best ways, and this was shown by those seminal studies back in 2009, is to do a tissue transplantation. And these seminal studies, a skin graft and an intestine graft were transplanted into new mice. And you can see that there are cells that will hang around within the tissue that is transplanted. Of course, recirculating T cells will come in to those new grafts, and those grafts will end up being a sort of a chimeric response of donor TRMs that were carried across in the graft, and also sort of old recipient TRMs that are being formed by circulating cells that are coming into that tissue. So you'll have both donor and recipient TRMs being formed within these transplanted tissues. And this has now been more recently, over the past couple of years, been shown to be true in humans. And this is a really nice study that came out last year from Donna Farber's group, who's in Columbia, New York. And she looked in patients that had received lung transplants over time. So here they would sample the donor who is providing the lung. And if you take the lung from this donor, transplant into a recipient, as I mentioned, you get this sort of almost chimeric response here, where the lungs will be made up of donor TRM, also circulating cells that are trafficking through the lung's highly vascularized, and also new recipient TRMs from this recipient, which are forming following transplantation over time. And if you look both in the CD4 and CD8 compartment within the lungs, and the way that you can identify these either donor or recipient TRMs is both by markers, and to tell if they're donor or recipient, you can use their distinct HLA types that would be present on the cells. And you can see here's this is looking many weeks. They looked up to over 12 weeks following transplantation, and you can see that quite clearly both CD8 and CD4 T cells from the donor lungs are still present within those transplanted grafts, showing that they're long-term and embedded within those tissues. The gold standard, which really has been used for mouse immunologists, has been parabiosis. And certainly across the last decade, many of studies have utilized parabiosis to show that TRMs are resident in pretty much every organ you can think of, or even compartments of the secondary lymphoid organs like spleen and lymph nodes also have a component of residency within those organs. But pretty much everything now, the lung, the salivary gland, tonsils, skin, genital tract, every organ of the sun has been shown pretty much to have a component of residency in there. And for these classic experiments, you would take a naive mouse that would be congenitally labeled. You would take an infected recipient that would have TRMs lodged within various tissues. Those resident cells would have a different congenic label, so you can see what cells came from the infected recipient, what cells came from the naive recipient. Once you stitch these mice together, cells in the circulation will go into equilibrium. So you'll have cells of the naive mouse and infected mouse in equal numbers between parabionts, but those resident cells will only be found within the previously infected site. And this has really been, as I mentioned, the gold standard for showing what is resident and what phenotypes are associated with resident cells by mouse immunologists. Now, another really nice way of showing that cells are resident are by doing, so in mice, people have used intravascular antibody injections, whereby you can take a depleting antibody or an antibody with a fluorescent tag, inject that into the mouse, and everything that's associated with the blood will be labeled. This works very well in the lung, which is highly vascularized, as I mentioned. Anything associated with the blood will be labeled with this antibody. The TRMs will be untouched. And also these studies have been very useful in human studies, specifically looking in the skin. So I thought I'd show you this really lovely example from Rachel Clark, who's a dermatologist in Boston, has been working on skin resident T cells for a long time. And here, if you just look at two different types of cutaneous T cell lymphomas in the skin, Rachel has denoted that there are those diseases that are mediated by pathogenic TRM cells, such as this condition here called mycosis fungoides. And this is where you get these fixed skin lesions that keep coming back in the same place at the same time. And if you go into this skin, it's littered with T cells of a TRM-like phenotype. And there are other T cell mediated disorders resulting in skin pathologies, such as leukemic CTCL, which is known to be mediated by T cells coming in from the blood. And here's the beautiful experiment that shows that. If you use alemtuzumab, which depletes cells expressing CD50, it's an anti-CD52 antibody, you can see that if you look at those diseases that are mediated by TRMs, before or after therapy, you don't have a TRM depletion. So you're not able to alleviate pathology. Whereas if you look at pathologies that are mediated by central memory T cells, you treat with anti-CD52, this wipes out T cells in the blood, and you can see that you get a resolution of disease here. Again, just highlighting to us, if we're thinking about T cell therapies, there are those where we want to target resident memory T cells, those where we want to target T cells in the blood, and you don't always play by the same rules. So you have to factor that in when you're thinking of therapies, when you're thinking about vaccines. Now, I want to spend a little time talking about this, and this is just how we define tissue resident cells by cell surface phenotype and by transcriptional profiling. And one of the sort of hallmark features of a tissue resident memory T cell is expression of CD69 by flow cytometry. Indeed, most tissue resident cells express CD69, but this marker is complicated because it's also upregulated by T cells upon activation. So it's not a true TRM marker because it's really a mixed bag if you just go in to tissues and look at CD69 expression. But certainly we know there's a core transcriptional signature of cells that are true resident cells. And this has been shown by quite a few groups now. And between all organs, both in humans and in mice, there's a core signature of cells that distinguishes resident cells from circulating cells. And our group was the first to show this back in 2013. And what we did was we embedded resident memory T cells in tissues induced by a whole array of infections. So we induced resident memory T cells in the skin using herpes. We embedded TRM cells in the lung using influenza, and those in the gut using a systemic pathogen that infects mice called LCMV. And then we looked at different populations in the spleen, effector memories, central memory T cells, and naive T cells. And if you throw the transcriptomes of these cells onto a PCA plot here, you can see the T cells that are resident band together and T cells in the circulation band together. So this shows you that those effector memory T cells actually have more in common with naive T cells rather than memory T cells that are embedded within tissues. And we pulled out a core signature here that was common to all resident memory T cells, and a lot of genes that came up within this signature, which were common to resident cells, perhaps not surprisingly, were those that were really modulating tissue egress. You need to shuttle those molecules down to get T cells to be embedded within the tissue. There was also a lot of upregulation of integrins, for example, important for tissue retention. So really modulating the egress and retention access to become a resident memory T cell is critical. So when you think about how a resident memory T cell is formed over time, you have naive T cells that are primed within the lymph node. And now it's actually been shown just last year that naive T cells might not all have the same potential to form either resident circulating T cells. So there might be some sort of fate decision very early which predisposes memory T cells to go down one fate route or the other. Now, those affected T cells that will proliferate up, they will go around the body, they will go into the site of infection. And once you get into the tissue, there's a whole slurry of tissue-derived factors which will condition a T cell to become a resident T cell. And those T cells will be left behind after infection for several months following infection. They'll express this core TRM transcriptome that I mentioned, and these T cells won't recirculate. So if you take that tissue, transplant it, or deplete cells from the circulation, these TRM cells will be untouched. So the differentiation program has been studied by many groups over the years, including ours. There's a lot of signals which need to come up and down, tissue-derived signals which are more or less important within certain organs. But something that is a kind of takeaway message, no matter what organ you're looking in, is the fact when an affected T cell gets into a tissue, you need to shut down that program of tissue egress. You need to downregulate molecules such as chemokines such as CCR7, that needs to be shut down. S1P molecules, you need to shut that down. Those that express those factors would actually go back into the circulation. Those cells that have shut down the tissue egress program will then sort of respond to tissue-derived signals. The microenvironment is very important in instructing the differentiation, and a lot of these signals are different between tissues, but the core transcriptome remains the same. So the first example that I gave you was T cells within skin, looking at these memory T cells that would be embedded within the tissue. And whether it's the skin or the gut, uniformly these resident T cells express CD103 and CD69 when you look at these memory T cells as opposed to their splenic counterparts. But now we know that if you look between different organs generated by the same pathogen, here we're looking at LCMV, we'll look more than 30 days later, gate on our transgenic CD8 T cells and look at the expression of these markers. Yep, by a different pathogen, exactly the same story for the skin, exactly the same story for the small intestine. But if you look in the salivary gland or the liver, you see a real mixed bag of expression of these surface molecules. And this has really led to an investigation for better surface molecules, so you don't have to rely on the transcriptomics to identify those cells that are truly resident in non-CD103 epithelial organs such as the liver. And this is something we've been working on for some time. We've been looking for new molecules that can identify resident memory T cells, and certainly molecules that have been studied over the years, such as transcription factors or adhesion molecules. If you look in different organs and you look at the clustering based on this expression, just the take home here is that they're different between organs, such as the liver and the skin. So this is very active amongst various researchers at the moment, looking for better molecules to identify these T cells. And again, using tricks such as parabiosis and tissue transplantation really to get at that. And certainly we've been pulling out specific signatures to specific organs. So I just want to leave you with the notion that there is a fair amount of heterogeneity within these tissues. Now, something that I really wanted to switch gears into is, again, showing you sort of, again, from the kind of earliest studies, why people are so interested in these tissue-embedded T cells. And that was the sort of first demonstration, along with the sort of recognition these cells existed, that these T cells provide superior immunity to peripheral infections as compared to memory T cells within the circulation. And this makes a lot of sense because these T cells are in fact positioned at the front line. So if you think if you've got a virus infection, if you have a virus here within a peripheral tissue such as the skin, you know, this is within a primary infection, you've got to, you know, the antigen's got to drain out of the skin. It's got to go to the lymph node. You've got to activate your T cells. They will come in. Of course, that takes a little bit of time. But if you even think about a vaccine that will just boost circulating cells and not resident memory T cells, you've got to wait for those T cells to wake up and come back into the skin. But you can see that if you would have T cells embedded at the right place at the right time, this might exert a more rapid control instead of having to wait for T cells to come in from other sites. So, and this is what T cells look like within the skin by 2-photon microscopy. They actually just don't move around a lot within that site. They stay very stationary when infection isn't present. But when infection is present, and these are just the tracks showing these T cells really are sticking around at the same place at the same time. But when infection is present, and I'll show this in a couple of slides time, these cells really wake up and start to get active. Now, I mentioned these T cells are present at sites of previous infection. They can be in, and what we know now, and this makes them attractive targets for vaccine strategies, they can indeed be embedded in tissues in a non-specific manner. And we know that they provide superior infection against infection compared to circulating T cells. Now, the very first study that showed this was, again, that 2009 paper that showed that TRMs exist. And this, again, was using this herpes simplex model. And very simply, remember, if you infect with herpes, you have a pool of resident memory T cells specifically embedded at the previous site of infection. Now, if you come back 30 days later, this is using a UMT recipient, so there's no antibodies in this mouse. So we're just looking at T cell mediated protection here. If you come back and you re-challenge this mouse, either at the previous site of infection or at another point on the mouse, what you find, if you measure viral titers a few days later by PFU, standard assay for looking at viral titers, you see at the left flank where you've got this pool of resident memory T cells, you have enhanced viral control. If you look at where you've got T cells within the mouse, because of course there will be memory T cells within this mouse, but you don't have that pool of resident memory T cells when we're looking on the right flank, if we challenge on that site, you get, of course, increased protection as compared to a control mouse, which hasn't been infected before, but you don't get the superior protection that you do when you have resident T cells. And this is really what kickstarted the field. We showed more recently, we formally would knock out either circulating cells or resident memory T cells to get a handle on what type of T cell does better. And with here, we used tricks where we could deplete either resident or circulating T cells. So just to take you through this, this experiment, here, we don't look at reinfection. We take a completely naive mouse. And what we do is we embed T cells specifically within the skin to form a pool of resident memory T cells. But what's important is that this pool of resident memory T cells is antigen specific. So they're specific for the virus that you're going to challenge with. And what you'll find after 30 days, you look at the markers, CD69, CD103, you get a pool of resident memory T cells expressing those markers. Of course, some of these T cells will leave to the spleen. You'll get a pool of circulating memory T cells as well. We can do tricks where we remove circulating T cells with various antibodies. So we've just got a cohort of mice here that has both memory resident cells and circulating cells. We've got a cohort that just has resident cells. And we also make a cohort where we don't have resident cells, but we've put back in circulating cells. So we've got a matrix here. We've got both, or we've got one or the other. And then we take these naive T cells that have memory T cells embedded at different locations. We challenge those mice for the first time with the pathogen, which those T cells are specific for, and we look for immunity. And here, just showing you the visual, we're looking at the site where we challenge. And what I can tell you is that when you've got TRMs embedded within that site, you get complete clearance of the virus. You don't see sort of this characteristic lesion appearing. But when you just got the circulating T cells here, they're not fast enough to come in and prevent herpes simplex. And you see this zoster-form lesion when you've got memory T cells in the circulation. And also what I can tell you is that that protection from infection given by resident T cells, perhaps not surprisingly, is stringently tied to the number of T cells that you have within the tissue. So the more TRM cells that you have, the decrease in viral load you will see. So very stringently tied to cell density. So I mentioned these T cells need to be virus specific, and this is just showing you this here, just using T cells that are virus specific here against herpes simplex, or using non-specific TRM cells. A lot of mouse immunologists like to use OT1s that are specific for chicken ovalbumin. So we've embedded TRMs that are virus specific or non-virus specific. We sort of give, we've challenged with virus here. The virus is cherry, so the virus is red. And you can see the virus specific T cells going into the lesion here and getting active, but these non-specific T cells completely ignore the virus and don't offer any protection at all. Our group and others have asked whether TRMs can directly kill infected cells. This is looking a little bit for a needle in a haystack. I'm still on the fence whether they can protect or cannot. But here is one image that we saw a few years ago showing that the HSV virus this time is in green. And here we have T cells in the skin here in red, and there's some evidence that these T cells can kill. But largely the way that the field thinks about how these TRMs can exert their protection is that they create an antiviral state in the tissue. And this was shown by 3 back-to-back papers in 2014 that were published in Science that showed that T cells within tissues could trigger a state of tissue-wide pathogen alert. So these TRM cells, when they see the virus, they can spit out a lot of cytokines, and in response will bring a lot of other T cells and innate cells into the site of infection. So just to sum this up in a cartoon here, what's thought when you see when a pathogen is encountered in the epithelium, for example, you'll get a resident memory T cell here. It might proliferate in response to infection. It might give rise to a new TRM, sure. But the first thing that it's thought to do is release a lot of cytokines. Specifically, interferon gamma is thought to be very important. And what this interferon gamma will do is that it will recruit NK cells, dendritic cells into the site of infection, also recruit other flavors of memory T cells in from the circulation, also B cells. And so it's really thought that these TRM cells are almost innate-like in that they can sound the alarm, recruiting in the troops when they see the virus first off to bring in a lot of other immune cells into the site of infection. So it's largely thought that they exert their functions through cytokine release. So something I thought I might touch on, and this is really only come to light within the past couple of years, is that TRM cells appear to protect against solid cancers as well. So in the past couple of years, it's been shown that cells that resemble TRMs, this hasn't been formally shown by those transplantation or parabiosis studies, but at least they have a phenotype which resembles a TRM. They've been identified in lots of different solid tumors over the past couple of years. And we showed last year that T cells can control melanoma within the skin. Here, showing here's a melanoma in red within the skin. If you've got embedded T cells within the skin, you get better control of melanoma. And again, we did our various tricks where we can either deplete circulating T cells or resident memory T cells. If you have control of melanoma and then deplete the resident T cells specifically, you can get outgrowth of tumors, showing that TRMs can promote cancer immune equilibrium. And just to show what we think this is looking like here, we think that TRMs that are embedded within the skin release a lot of cytokines, keeping these T cells within check. And also it's been shown that T cells are associated with improved prognosis against various cancers. And just to show you an example here, this is going, if you look at patients with breast cancer, this is just one example, several examples have come out in the literature over the past couple of years, and you just sequence the CD8 T cells within looking at those TILs within breast cancer tissue. And if you look at these CD3 positive T cells, there are different subsets of CD8s within here, but there is this cluster here and the clusters together that has a signature which resembles the TRM signature, which was found, that core transcriptional signature that was found within mice. And if you take this signature and you look at the prognostic, how people do, depending on whether they've got a high density of these cells or not, it's actually had very powerful prognostic capacity, just comparing it to whether you had T cells or not, or even Tregs or not. The overall survival correlated best with those patients that had a lot of TRM cells, indicating that patients that have a lot of resident memory T cells within tissues might do better against cancer. And again, what we know about TRMs compared to their counterparts in the circulation is that they have extremely high expression of immune checkpoints, such as PD-1, even though it's thought because they can proliferate and function very well, it's thought they're not exhausted, but they do naturally have this naturally high expression of checkpoints. And again, in patients that could respond to anti-checkpoint blockade therapy, such as anti-PD-1, responders had an enrichment of a TRM signature here. So what I've shown you, and here are just some takeaways, is that TRM cells are a non-recirculating population of T cells that are found within tissues. I've shown you that TRM cells have a distinct development program that induces these cells, and they have a distinct transcriptional identity, which can be used to identify them, comparing them to circulating memory T cells. There's a lot of heterogeneity within the TRM populations in different tissues. This, perhaps not surprisingly, is really dictated by the different tissues and signals available within that tissue. And indeed, there are different requirements for T cells within different sites. And I've also shown you some evidence that TRM cells mediate local immune protection. It's been shown this is true against a variety of infections, but also what's coming to light is that they're also important in various malignancies. And so new therapeutics that incorporate memory T cells are now really being thought of. So I thought I would just finish up by coming back to my first slide, just thinking about the sort of protection that we would want from a vaccine, for example, against SARS-CoV-2. So of course, antibodies are going to be incredibly important, but also it's been shown that T cell responses against COVID are also very critical. And perhaps we should also be thinking about the type of memory responses that we would want to induce. We know that the type of response, the type of adjuvants or inflammation, the types of affinities of TCR, there's different rules. If you think about, and also the route of vaccines, the induction of circulating T cells versus resident T cells, they play by different rules. So strategies that might induce a high number of circulating cells might not give rise to a population of resident T cells. And here what we would be looking for is T cells specifically in the lung, which really might be on the front line when we're thinking about a respiratory virus. So we know that the immunity that we would want to induce, we want tissue resident memory T cells that might be in the lung, or within the bowel, within the nasal cavity. It's been shown they're highly protective against respiratory infections such as influenza in mice. But of course, we also will want to induce circulating memory T cells that can come in high numbers and help to clear that infection. Now, to develop a resident memory T cell, just to remind you, I've shown you there are distinct requirements. Effector T cells that get into the tissue in the first place. There are factors within the tissue that are important. And then you get a whole lot of changes specifically on this resident cell. Upregulating molecules important for tissue retention. Molecules will be downregulated that prevent tissue egress. But those T cells that will be embedded when you see a pathogen for the first time, if these T cells were embedded by vaccination, when a virus will come in, say to the lung, for example, these resident memory T cells would secrete a lot of cytokines in response, primarily those such including interferon gamma. And upon secretion of those cytokines, that would help facilitate a very rapid immune response, whereby you would get enhanced recruitment of macrophages, also dendritic cells. The dendritic cells will, of course, then take up the antigen, go to the lymph node where they might wake up central memory T cells that could be reactivated, come back into the site of infection. And of course, we will also have our circulating effector memory T cells or effector T cells that will always be able to come in straight from the blood in response to infection, and potentially having both these resident and high populations of these effector T cells might lead to the best immunity within the lung against respiratory viruses. Right, so I'll close there. Hopefully I took you on a bit of a history through sort of, I guess, the sort of foundations of learning about resident memory T cells. Also shared some data from my group. So just to acknowledge my team in Melbourne and our funding sources, also collaborators. And I'd like to thank everybody so much for your attention in this talk, and also the organizers for giving me a chance to present. And thank you again.
Thank you. We have 15 questions. I don't know whether we'll have time. So, Professor Aroras, do you think we can entertain all the questions, hopefully cutting the break time a little bit?
You can pick up 4 or 5. There are sometimes repeats, or some you can club them.
Okay, okay. First question was that although malaria has been around for so long, why there is no vaccine developed against malaria?
That's for you, actually. That's for you. Question is for you.
So we always answer this because it's a multi-stage infection. It is so complicated. We are yet to understand exact immune response against various stages of malaria. That is a typical answer we give, but although I encourage my students not to believe in that, but we still feel that we can develop. At least whole sporozoite vaccine has taught us that it is possible. But similarly now, Michael Good in Australia is talking about whole blood stage vaccine, that also. So I'm sure now, because I think most of the efforts have not been concentrated for this, because time to time we have new drugs come. But now efforts are on. I'm sure in next 10 years we'll have vaccine against malaria. So at least start with whole sporozoite.
And something that I should point out regards to the efforts in developing a malaria vaccine is that there are certain groups out there, including Bob Cedar at NIH, that are really considering getting high pools and high densities of liver resident T cells to fight the liver stage of that virus as well.
Yeah, yeah. So because that is, they think this is that liver resident T cells are key. This is ISD 69 required for TRM cell formation. CD69 deficient CD8 T cells exhibit reduced TRM cell formation in skin, whereas CD69 negative CD4 T cells can generate TRM cells in lymphoid tissues. So would you like to comment on this?
Yes, that's a topic that's very close to my heart, because it was our work that showed that CD69 was important for skin, and it's a close collaborator of ours work, Steve Jameson in Minnesota, who showed that wasn't true of other tissues. And this is again coming back to different T cells in different sites have different requirements, and they play by different rules. So in the skin, it's very stringent what you need to shut down in terms of tissue egress. And CD69 has a reciprocal relationship with S1PR1, important for tissue egress. And indeed, it's required for the skin, the lung, the gut. You can knock out CD69 and those T cells are absolutely fine, but you still need to downregulate S1PR1 to shut off the egress program. They express CD69 still, but they don't rely on it.
So is it logical to apply mouse memory T cell findings to human memory T cell? There is a lot of differences coming up.
Yeah, there are indeed a number of differences coming up, but there's also a lot of similarities. And say, if you take the core transcriptional signature, which was developed in mice, it's reciprocal in humans. Also, the transplantation experiments in humans have really really beautifully recapitulated what was shown in mice so long ago. And without the mouse models, we just wouldn't have known what to do in humans, because it really kind of took a huge effort in investigating tissue resident memory T cells in humans, because both our lab and Donna Farber's lab in New York had to set up a donor program where you actually have to to isolate these T cells, you actually have to take tissues from deceased donors to really get enough to look at them. So a mouse starting point, but there are a lot of similarities coming out. Now we're able to investigate in the human space.
How about any homeostatic crosstalk going on between circulating effector T cells and resident tissue T cells in health and disease condition?
So Certainly, I can tell you that you know, if you have an active site of infection or if you have inflammation, you will have both effector and memory T cells in there. It's just when all inflammation is gone, you will only have the resident T cells. It's known that you get enhanced infiltration of circulating memory T cells if you do have resident memory T cells, so there's that interaction. But it doesn't seem to be the other way around in the fact that you need circulating CD8s to have resident CD8s. But what has been shown recently, at least in some tissues, and this example is the genital tract, you need CD4s to induce a good population of CD8 resident memory T cells in the first place. But again, I issue caution that it seems to be different between different tissues.
So what kind of changes at cellular level makes memory T cell differ from normal T cell? From cytoplasm, DNA, receptors, you know, yeah.
So CD8 T cells, the memory T cells, they certainly switch their metabolism. So they're more reliant on fatty acid oxidation, for example. They start to switch their cytokines, which they need for homeostatic proliferation. They seem to be more IL-15 dependent, whereas your naive T cells are more IL-7 dependent. They're more long-lived. Certainly the central memory T cells, they have rapid effector function. Certainly the effector memory T cells and resident memory T cells compared to naive T cells. So there's a lot of changes that happen between the naive and memory T cell pool.
Any chemicals that can increase TRM cells in circulation reported?
Certain chemicals, I mean, something we are investigating at the moment is certain small molecule inhibitors and compounds to induce resident memory T cells. That's something that we're looking at with various pharmaceutical companies. So it's definitely possible. That's an ongoing work, but nothing's been published as yet.
Are TRMs seen in immunological privileged sites?
Yeah, it's been found that they are found in the cornea and also within the brain. And how they're able to cross the blood-brain barrier is something that I'm not sure about.
How resident T cells can prevent autoimmune disease like SLE and multiple sclerosis?
Well, I don't think they prevent it. I think they can potentially, it's touted, at least it's thought that potentially they contribute by having all these T cells embedded within tissues, that they can be at the wrong place at the wrong time and start to misfire. But it's only really been well displayed for skin disorders like psoriasis at this time.
Somebody has asked this, can macrophages mimic memory cell-like effector function?
I think I would say that their functions are quite distinct. I mean, I guess they're both good for providing immunity against infection. That's the similarity they have.
So please throw some light on CD4 tissue resident memory T cells.
Yeah, absolutely. So CD4s have come a little bit later in the literature. CD8s were studied very heavily to begin with, but it's now been shown that CD4s can make good residency as well. It is sort of debated in the field whether they are as long-lived, even though they can put on the residency program. They've shown to be resident by tissue transplantation and also by parabiosis, but it's thought they don't live as long as the CD8s.
There's a question, what is the role of TIL in human cancers? Yes, please.
Yeah, so it's known that TILs can be important in, at least if they're not exhausted, in creating a good immune response against cancers. It's highly prognostic. If you've got a high number of TILs, you're likely to fight that cancer more effectively. And it's now thought that a portion of those TILs are going to be our resident T cells.
So you think all TILs are not TRM?
No.
So, Dr. Sarath, can I ask a question from related to the cancer only? What I understood from your talk, Laura, that these tissue resident memory T cells are antigen specific, driven by some pathogen, then you have a T cell response. You get those T cells go to resident tissues and they stay there, and they are tissue resident memory cells. They are carrying some signature. I mean, they are carrying some memory about a antigen, right?
Yeah.
So then you said they protect you from melanoma. What is this antigen specificity? Are they cross-reactive, or how do they protect the cancer when they are not seeing the?
Yeah, that is a really great question. Something that's absolutely so active in the literature at the moment, because it was only a couple of years ago, really, that they were identified within the tumor microenvironment. Now everybody is looking for antigen specificity. It's the school of thought is that there's going to be T cells in there that are melanoma specific, but there could be some cross protection by bystander T cells, and it's also it's kind of been shown that if you embed virus specific T cells, you can get some protection, at least in mouse models, against various cancers. But I would think there has to be an element of antigen specificity in there to get protection against cancers.
So,
and how these are arising, whether you just have a little bit of sort of antigen, and then that pool sticks around, controls the cancer, and then it gets bigger, and there's this equilibrium of control and so forth. I think that might be how it's working.
It has to be some kind of antigenic, cross-reactivity. Otherwise, it's not the statement of that these tissue-resident cells will be protective against any cancer may not be true, right? It has to be specifically related to some antigenic cross-reactivity, right?
Yeah, it's a highly debated issue at the moment.
So what do you know about the longevity of these TRMs? And can we induce them by vaccination? Yes.
So it's been shown, at least in the skin, they're incredibly long-lived. So if you do a facial transplant, for example, you can find skin-resident memory T cells that last for years within the donor. So they're thought to be very, very long-lived. Those in the lung, in comparison, are thought to be less long-lived. So it's thought that those in the lung, you need repeated boosting by antigen to keep them there. They can stay a few weeks, but it's shown that they decay. So it's thought, again, it's different between different tissues. They are still long-lived, but some are more long-lived than others. And those in epithelial are the most long-lived.
Does TRM play any role for chronic bacterial infection like TB? Or is it more specific for viral infection?
No, it's been shown that TRMs can also protect against viral infections such as leishmania. And against, I know at least one group who's working on the role of CD4 resident memory T cells and TB at the moment. So it's an active area of investigation.
How about overreactivity of skin resident T cells in autoimmune skin diseases like psoriasis? Would you please comment on the origin of stimulating signal, systemic versus local? Yeah, so at least the evidence at the moment is that TRMs are in high density in sites with psoriasis. They have a TRM signature. They're producing a lot of IL-17. That correlation, it's only the correlation that's been shown at this point in time. Whether they, to show that they formally do it to deplete them out, I don't think it's been shown well at this point in time. But certainly the correlation that they're there in those sites is in the literature now. Are vaccines available for fungal infections, and why they reoccur again and again? Vaccines for what, sorry? Fungal infections. Fungal infections. Don't know if I can answer that one. I mean, something that I can answer. I don't think I can answer that one. It's okay, tuberculosis has been done. Does microbiome have any influence on resident memory T cells? That's an excellent question. I know quite a few groups working on that at the moment. Yes, and we are considering those natural epithelial lymphocytes in the gut intestine to, they've got a lot in common with resident CD8 T cells. And certainly the microbiota is very important in inducing that flavor. And certainly even if we associate the skin with a commensal bacteria, it can really change the function and flavor of memory T cells, and it can induce more of them. would a transplantation of tissues containing resident T cells provide an advance or improve response in the naive host?
No, I thought that it's meant to be worse. It's kind of it's thought that generally when you do a transplant and you transfer over those donor T cells, you don't really want that, because it's thought that those donor T cells might reactivate and start attacking the host tissue. And actually, in that facial transplant example that I mentioned, 12 years later, these donor T cells in the skin woke up and started causing pathology in the tissue, and that's how the sort of pathologists picked up that it was resident T cells that were causing pathology. So I don't think you want them.
It's okay.
So we need to wind up now.
Yeah. So thank you. It's a very exhaustive session for you. So I'm sure if anybody else has any question, they can email. Dr. Arora, we should be able to provide the email ID to all the participants.
Yes, we will like to
Some
share.
have asked questions, and also some have asked question if they can get the PDF version of the PPT. Will that be?
We'll
Yeah,
have to get consent from all the speakers, and we'll try to. Although we are recording it, so the recordings will be available on website, but PDFs providing is I can't say something at the moment.
Okay.
No problem.
So
Thank you.
Yeah.
Pleasure.
About how much? Maybe about 10 minutes break. We get back again and start another session at 12 o'clock. It's a short break. Thank you very much.
With seminal discoveries regarding basic knowledge and clinical study of NK cells, particularly liver-resident NK cells, cytokine-producing NK cell subsets, and NK cell-based immunotherapy. He has published more than 300 papers in peer-reviewed journals. So over to you, Dr. Tian.
Yes. Yeah. Hi. Can you hear me?
Yes, we can.
Yeah. Okay, thank you. Thank you very much for your introduction.
Thank you, sir.
And okay, so let's move to the next session. And we have the two lectures. And the first one is Professor Amida Avashi. He is an associate professor of the Immunobiology Laboratory, Translational Health Science and Technology Institute, Faridabad, India. Amida Avashi did his MS in biotechnology from Jivaji University, Gwalior, India, and the PhD from the National Center for Cell Science, Pune. He had his postdoc training at the Brigham and Women's Hospital and the Harvard Medical School, Boston, Massachusetts, USA, and worked as instructor at the Harvard Medical School, Massachusetts, USA, before joining THST Faridabad as faculty. His present research interest lies in the area of understanding the interplay between effectors and regulatory T cells in IBD and gut infection, and the metabolic regulation of T cells during their differentiation. Besides, his lab is trying to identify and understand the role of checkpoint inhibitor in cancer immunity and understanding transcriptional regulation of Th9 and the Th17 cells. So today, his lecture title is T cell subsize and cytokine storm. Dr. Amida Avashi, welcome you to give us lectures.
Thank
Can
you.
you hear me?
Thank
Yes,
you.
yes.
Thank you,
That's
Jagan.
good. Yeah. Very happy to see you, my
Yes,
friend.
it was great to see you last year in China. So
Okay, thank you.
I can just share my screen. Yes. So basically, I'm going to go through stuff which is like T cell subset, and just quickly see. Okay.
Yeah,
So
could you
T cells-
please to have the full screen? Full screen. No, it is not the full.
Okay.
Yes. Okay, thank you.
T cell subset, and as we know that effector T cells, and I mean T cells can be differentiated into effectors as well as regulators, And what we are talking about here is the precursor cells, or we call it naive T cells, or we call it virgin T cells. And these T cells are the one who has actually not seen any antigen. So they are kind of naive T cells. And the moment they see the antigen, they differentiate into different subset of T cells. Like for example, when they see an antigen and at the same time they see the cytokines like interferon gamma and IL-12, they become Th1 cells. And these Th1 cells are actually developed by a transcription factor STAT4 and Tbat. And the Th1 cells produce a massive amount of interferon gamma and are very crucial for elimination of intracellular pathogens. And these cells together with Th17 cells, or maybe these double producers, what we call IFN gamma and IL-17 double positive T cells, are kind of play a major role in inducing autoimmunity when they are actually sees the autoantigen. The same naive T cells can differentiate into Th2 cells with IL-4, and there is a separate transcription factor for this differentiation is GATA3 STAT6. Th2 cells produce IL-4, 5, and IL-13 and are very important for clearing extracellular pathogens like worms through IgE-mediated degranulation of mast cells and other granulocytes. So these cells are important for extracellular pathogens. However, when they are dysregulated, the Th2 cells can actually induce allergic inflammation like asthma. In addition to Th1 and Th2 cells, in 2006, the differentiation factor for another subset of T cells has been identified, demonstrated that TJ beta together with IL-6 induces differentiation of T cells, which is distinct from the Th1 and Th2, are Th17 cells, and Th17 cells having a different and very distinct transcription factor than Th1 and Th2. This ROR gamma T and STAT3. Th17 cells produce IL-17 cytokine and IL-22, and primarily important for clearing or elimination of extracellular pathogen like fungal infection, which normally Th1 and Th2 cells can do. But beside clearing the fungal infection, the primary or predominant role of Th17 cells has been described and demonstrated very nicely in the literature, causing autoimmune disease and particularly organ-specific autoimmune disease. In addition to Th1, Th2, Th17 cells, Th9 cells has been also described, which is can be induced by TGB and IL-4, and these cells produce IL-9 and having a different transcription factor. And my lab has been shown that back in US, we have identified these cells in Vijay Kuchru's lab, and then I could keep continuing working on Th9 and Th17 cells in my own independent lab. And we have identified a transcriptional regulation of Th9 cells and found that in addition to the transcription factor, which is IRF4 PU.1 STAT6, we've shown that FOXO1 is also important for IL-9 induction and Th9 differentiation. And the role of Th9 cells is actually very similar to Th2 cells, and that's what we call them a sister population. And this Th9 cells induces when they dysregulated, it causes asthma and allergy, but major role of Th9 cells, which is coming in the literature, is inducing anti-tumor immune response, even much more potent than the Th1 cells. So this is what about the effector cells. On the other hand, you have the regulatory cells, because in order to control these effector cells, you have to have something in the system at the cellular level. These are the regulatory T cells, and regulatory T cells primarily you can actually divide into two major regulatory cells. One is the FOXP3 positive regulatory T cells, and within the FOXP3 positive regulatory T cells, you can see there are two classes of regulatory T cells, or Treg cells, the FOXP3 positive and Treg cells, which is naturally occurring Treg cells. They are actually originated in the thymus at the time of positive and negative selection. At the same time, when the T cells actually come into the periphery, they can also be induced by a cytokine called TGF-beta, and these cells called induced Tregs or iTregs. They are kind of very similar in inducing suppressor function and control these effector cells. But there are different markers on these cells which can be by which you can identify which one is actually an Tregs or which one is the iTregs. In case if somebody is interested to see heat mapping of Treg cells. TH3 cells came out for some time, but there is no molecular description has been documented. Therefore, it's kind of dried out. And the other cells, which is coming up very nicely, is the TR1 cells, which initially was identified by Maria Gracia Ronacarlo, that TR1 cells produce massively IL-10. And these cells are FOXP3 negative. So these cells don't have FOXP3, but they produce massive amount of IL-10. And this IL-10, you know, that is an anti-inflammatory cytokine, and that can control the immune system in various ways by controlling DC function, by controlling T cell proliferation, and so on. And these cells are what we have identified. The initial discovery in I think early 2000. That IL-10 could be a potential differentiation factor for TR1 cells, but later on what we have identified in 2007, that IL-27 instead of IL-10 is the major driver for inducing TR1 cells and produces IL-10. So basically, after the discovery of Th1 and Th2 paradigm somewhere in 84, 80, 85, 86, by Bob Kaufman and Tim Mossman, the kind of T cell differentiation field was kind of very much dried, and nothing was really happening at that time. Only thing what happened during this time after the discovery of Th1 and Th2 cells, that every single immunologist was trying to boxing the disease, whether it's an autoimmune disease or whether it's a infection, either into Th1 box or Th2 box, right? So something associated with the Th1 cells and something which is associated with Th2 cells. And other than that, there was not really much was happening in the field of T cell differentiation. So there is some kind of current in the water started at the time. People are more interested to see the tolerance mechanism, how T cells are being tolerized and how our body is actually having this immune homeostasis. So this is actually, I have taken from a review where Thymus derived T cells. So if you have a positive selection and negative selection, certain cells actually coming out of this, and they induces a kind of tolerance. So I briefly mentioned to you these Tregs, N-Tregs and I-Tregs. N-Tregs are generated in the thymus. I-Tregs are generated in the periphery from the non-Treg population. And these Treg cells express a transcription factor, which you can really very nicely isolate these cells if you have a kind of tools, FOXP3 positive T cells. And the mechanism by which these Treg cells actually suppresses the immune system is contact dependent as well as contact independent, but primarily it is contact dependent. Other than the Treg cells, the TR1 cells, which is type 1 regulatory T cells, induced by IL-27 and produce IL-10 and induces the immune suppression in IL-10 dependent manner. So this T cell tolerance field was kind of very much alive at the time, 1969, when there was a kind of, later on, it has been shown that neonatal thymectomy of a normal mice between day 2, day 4 after the birth results in the destruction of ovaries. So initially, when this experiment came out, it was thought that maybe the thymus is producing something which is important to take care of the ovaries. Maybe some sort of hormones are being produced by the thymus. So it was an observation, was a very nice observation, was really not really connected to the level of immune system. And later on, depending on the mouse strain used, this thymectomy, which is like also called 3-day thymectomy, because it is most efficient if the thymus is removed day 3 after birth, lead to the development of various thyroiditis, gastritis, orchitis, psoriasis, and various kind of diseases. So it looks like there is kind of correlation in thymus and all these conditions if you remove thymus. That means the thymus at day 3, there's something is being produced, or maybe some cells are being produced, which is really controlling these conditions. If you somehow remove the thymus early days after the birth, these cells may not be generated, and therefore immune homeostasis is kind of broken. And later on, other discovery by Penhale et al. Reported that adult thymectomy, which we called ATX, of normal rat, followed by 4 rounds of bi-weekly sublithical X irradiation, produced autoimmune thyroiditis. So it's not only in the neonatal thymectomy, it is also in the adult thymectomy, followed by the irradiation. You also get some sort of, that means the thymus you are removing in the adult stage, but whatever the cells are being produced by the thymus during this time, if you irradiate them, that means then the cells which is kind of inducing tolerance are completely gone, and thymus is not there. So therefore, like autoimmune kind of disease is being produced in this settings. And later on, it has been shown CD4 positive T cells and CD4 positive CD8 negative mature thymocyte inhibited this neonatal thymectomy. So basically, if you actually remove the thymus in the neonatal setting and you come back with the thymus cells, which is CD4 positive and CD8 negative, and put back into the mice, these mice actually getting better. There is no sign of any disease. So look like this proportion of CD4 positive cells from the thymus containing something which is actually controlling the disease. And this is actually by a very nice paper. I think Sakaguchi has shown very nicely that during at this time, these regulatory cells or Treg cells actually being produced. So this is the optimum time to remove the thymus. And then you can have this normal mouse. You can put back regulatory T cells, and you can actually control this autoimmune disease. There is no disease when you bring back these CD4 positive, CD8 negative T cells. But the problem was in the field, and later on it has been identified this CD4 positive cells contains a pool which is called CD4 positive and CD25 positive cells. And that's how they have identified a marker which is a CD25 on the surface. And the CD4 positive, CD25 negative, we call them effector cells. And CD4 positive, CD25 negative cells, we call them regulatory cells. This is after the discovery of Sakaguchi classical paper on this. But the problem with this system that if once that normal T cells, which is effector cells, is getting activated, right? So when they are not activated in the normal immune homeostasis, when the immune system at the natural state, there is no infection or autoimmune disease is happening, you have this effector and regulatory very clear-cut distinction. But at the time when there is an activation of immune system by some sort of infection, virus, pathogen, bacteria, whatever you call it, these effector cells start expressing CD25 on their surface. So now at this stage, when the immune system is activated, you cannot really distinguish between the effector cells and the regulatory T cells based on the CD25 surface expression. So this was a time that people really wanted to have some distinct marker or some distinct transcription factor to distinct these effector and regulatory T cells to go into more detailed analysis of regulatory and effector T cells. So there are three seminal paper actually came in two was in Nature Immunology, one in Science, one from definitely from Sakaguchi group, and one from Sasa Rudensky. So this took almost like close to 20 years that after the discovery of CD25 positive cells in 1985 by Sakaguchi and later in 2003, the transcription factor has been identified for these cells, and therefore now we call them FOXP3 positive, CD4 positive, C25 positive cells. And after this discovery of transcription factor, people has made them knock-in mice in which the FOXP3 gene locus is actually having the GFP into it. You have the GFP under the locus of FOXP3. So at any time the FOXP3 is going to be induced, the GFP, you can label these cells with the green. So this gives us a very nice tools to actually induce to study the FOXP3 positive regulatory T cells. This is 2003. So this Th1, Th2 paradigm, as I mentioned to you, Tim Mossman and Bob Kaufman has done it, and the GI paper, and I think it's a kind of citation might have been increased. It's an old slide. And he said that they have described naive T cells can become Th1 and Th2. Th1 can take care of the intercellular pathogens. Th2 can take care of the extracellular pathogens. But I mean, they're kind of some sort of, and this is the paper I was actually talking about, they have actually mentioned. So they have identified this clone. This is a Th1 clone and the Th2 clone. This is the original data from the paper. And the Th1 clone is actually making tons of interferon gamma, and Th2 clones is making tons of IL-4. And that's how these clones, and they're kind of mutual exclusive, you know. So once you have Th1 interferon gamma, they cannot really have Th2. And once you have the Th2 cytokines, the Th2 cells cannot produce Th1 cytokines. And later on, it has been identified how these antagonism happens. So interferon gamma suppresses IL-4 and IL-4 suppresses interferon gamma. So as we know that Th1 clone, what he has identified induces plasma cells, and these plasma cells produces IgG2A, IgG3. Although IgG2A is not really shown in terms of human settings, but this is very well true where anytime you like to see the Th1 response in terms of antibody class switching, obviously everybody like to do in mouse setting to do IgG2A. Similarly, the Th2 clone through IL-4 induces IgE, as we know that Ig is important for allergic inflammation, and it induces mast cell degranulation through Fc epsilon receptor. So now, for example, how these Th1 cells are generated, I just, for the sake of student, I just say that this pictorial depiction that pathogens actually comes and they interact with the dendritic cells through the toll-like receptor, which has been taken care yesterday, how toll-like receptor is actually activates the dendritic cells. So once the toll-like receptor get activated through these pathogens, the dendritic cells produce interleukin 12, and these antigens are actually being shown to TCR through the class 2, and these naive T cells, which express IL-12 receptor, which induces which induces interferon gamma production, and this interferon gamma in an autocrine manner actually further amplify the Th1 differentiation. So this is like a kind of summary of how the Th1 cells being induced and differentiated. So this is how IL-12. So if you go back to IL-12 receptor and IL-12 signaling, so IL-12 is a heterodimeric cytokine, is composed of P35 and P40, and two together make bioactive IL-12. The receptor of IL-12 is also a heterodimeric receptor, is IL-12 receptor beta 1, which is non-signaling unit, and IL-12 receptor beta 2, which is a signaling unit, makes IL-12 receptor. And once IL-12 bind to IL-12 receptor, it induces STAT4 activation through the phosphorylation. And the STAT4 actually further goes into the nucleus and activate the Th1 programming. So for example, if you, each of these things has been proven experimentally. For example, if you make IL-12 knockout mice, whether this is IL-35, P35 this unit or P40 this unit. These mice are susceptible to mycobacterial infection and they don't have Th1 response. Similarly, if you knock out IL-12 receptor beta-1, this subunit, or IL-12 receptor beta-2, these mice again also susceptible for infection and there is no Th1 response. And similarly, if you make a knockout of STAT4, and which is also very similar to IL-12 and IL-12 receptor, suggesting that each of these component of IL-12 signaling is really crucial for controlling Th1 response. And if you don't have Th1 response in the absence of either of this, there's a kind of susceptibility to the intracellular bacterial infection. So very similarly, so once IL-12 is signaling, the IFN gamma comes in to further amplify the Th1 response, which I've shown. And this interferon gamma is a cytokine which induced by T cells primarily, bind to IFN gamma receptor 1 and 2, this again heterodimer. IFN gamma receptors signaling induced JAK1 and JAK2, and this JAK1, JAK2 induces the activation of STAT1, and this is STAT1 further amplify the Th1 immune response. So again, these things has been shown at the level of knockout settings, that if you make IFN gamma knockout or IFN gamma receptor knockout or a STAT1 knockout, there is no Th1 response, and these mice are susceptible to intracellular pathogens. So now these things together actually makes a point that both IL-12 and interferon gamma is important for Th1 differentiation, as well as important to clearing the intracellular infection. So now I put everything together to make more sense in a kind of a pictorial manner. So basically what I have done, I have divided the Th1 differentiation in two parts. One is the induction part, and second one is the amplification or stabilization part. So the induction part happens in context of dendritic cells activation through the TLR, right? And the IL-12 is being produced because this is primarily produced by the dendritic cells or antigen presenting cells, which induces STAT4 and it binds to IFN gamma locus, induces IFN gamma. Once IFN gamma is produced, this IFN gamma goes back and induces its own amplification through the IFN gamma receptor STAT1 pathway to have more activation. So now, so far so good. I mean, the STAT1, STAT4 and all this kind of things looks fine, but at this time, people were really interested to identify the master transcription factor of Th1 cells. So the lab in the Harvard, Laurie Glimcher's lab, was really interested to do this kind of work, and she did a lot of screening of expression cloning and identified one of the protein which is primarily expressed in the Th1 clone, not in the Th2 clone. As you can see the data here, this is from the Cell paper, that any time, whether day 1, day 2, day 3, day 5, day 8 differentiation of Th1 cells, you can see a lot of Tbat expression in each of these days, but this Tbat is not actually coming in Th2. Remember, this is a time at 2000, there was no Th17 cells discovered at that time. There is no FOXP3 positive Treg cells were discovered at that time, right? The only thing what people were really wanted to know molecularly, what is the role of Th1, Th2 cells and how they are molecularly can be defined. So Tbat comes out as one of the factor which is important for Th1 differentiation, and this actually bind to IFN gamma locus very nicely, and they have shown it by the luciferase assay that anytime you put luciferase IFN gamma together with the Tbat, there's a nice luciferase activity. And when you actually overexpressed, retrovirally overexpressed Tbat into naive T cells and activate them, these cells start producing massive amount of IFN gamma at any time point. And during this time, the Tbat, later on, it has been shown that Tbat can suppress Th2 cells and bring Th2 cells into Th1 cells. So Tbat is having a capacity to bring back already differentiated Th2 cells into Th1. So now the model looks very kind of complete. The induction induces STAT4. The amplification through interferon gamma also induces STAT1, and both of them actually merge at the major transcription factor TBAT. And if you don't have TBAT, despite having the whole system intact, like this whole pathway and this pathway is intact, but if you don't have TBAT, there is no differentiation of Th1 cells, there is no interferon gamma. So therefore, we call it a master transcription factor of Th1 is the TBAT, and it's called TBX21 as a gene name. So far, I have actually covered that IL-12 induces interferon gamma into Th1 cells. Transcription factor STAT1 and STAT4 is also important for development of Th1 cells. And transcription factor TBAT is the master transcription factor for Th1 cells. Th1 cells are crucial for cell-mediated immune response against intracellular pathogens. But in the literature at this time, there was evidence that interferon gamma producing Th1 cells actually accumulated in the lesions of MS patient, and reactivation of cerebrospinal fluid T cells with the myelin antigen can produce interferon gamma, and exacerbation of multiple sclerosis in the patient treated with recombinant interferon gamma. So these evidence actually implies that interferon gamma is somehow important for causing the disease severity in MS. So MS or multiple sclerosis can be studied in the very nicely in the animal model. This is called experimental autoimmune encephalomyelitis, EAE, a mouse model of multiple sclerosis. And the model has been generated in a way that if you actually take the myelin antigen and if you emulsify them into CFA, those antigen could be MOG, PLP, MBP, and these, if you emulsify and immunize the mice get paralyzed around day 12 to day 15. And this is how the scoring system, this is like score 1 naive mice and preclinical score 2. At onset, you have, this is onset, at the peak of the disease, score 4. And this is a very fairly nicely robust scoring system, which anybody can do. It's kind of very easy to do. And when you actually take the brain or CNS of the mice at a score 4, you can see there's kind of lesions in the brain as compared to naive. So this is the kind of system people use to study antigen-specific T cell response in a very precise manner. And when you immunize the mice, they look quite kind of paralyzed. They cannot really move in the cage. You have to provide food and water to them, and actually you have to pay extra attention to them. So when people are talking about Th1 cells could be important for inducing autoimmune diseases, there are kind of some paradox in the literature was really kind of bothering immunologists, because if you just see all these Th1 molecules, and this is I'm talking about at that before the discovery of Th17 cells. So all these Th1 associated molecule, if you immunize them with the EAE, some of them get paralysis and some of them don't get paralysis. Right. So this actually brings all immunologists into a conclusion that there's something is really not correctly interpreted. Right. So to come in to this, there was a discovery of IL-23 and IL-23 receptor. This is the time again 2000. The paper came in Immunity, identifying a novel P19 subunit, which combined with the IL-12 P40. Remember, I told you that IL-12 is composed of P40 and P35. So now P19 can also bind to P40 to make a novel cytokine, which is called IL-23. And 2 years later, the receptor for IL-23 has been, so initially when this discovery happened, people thought maybe this is also kind of going to induce Th1 cells because it is P19 combined to P40. Maybe it's going to be having some association with the Th1 cells. But later on, the receptor for IL-23 is also being identified, which is also kind of heterodimer. And this is actually combines IL-12 receptor beta-1. Remember, I told you that IL-12 receptor is having two subunits. One is the beta-1 and one is the beta-2. So now IL-23 receptor can combine with the beta-1 to make a functional receptor for IL-23. So this was the kind of complicated system which you can see now here. that IL-12, you have 2 subunit, IL-23, you have 2 subunit, the common subunit is P40 between them. IL-12 receptor is having a 2 subunit, IL-23 receptor is having 2 subunit, but the IL-12 receptor beta 1 is the common subunit. So now if somebody has done the experiment using P40 knockout mice or IL-12 receptor beta 1 mice, which I've shown you in the previous slide, those knockout is not necessarily telling you that this is because of Th1. It could be because, sorry, this is because of IL-12 or IL-23, because it could be the phenotype what you see in this knockout, particularly P40 and IL-12 receptor beta 1 could be because of IL-12, because IL-12 will not be functional here, and it could be because IL-23, because IL-23 will not be functional here either. So how to solve this paradigm, I mean, how to solve this paradox or the complication? Only way to solve it to make IL-23 receptor alpha knockout or IL-23 receptor P19 knockout. Then only you can say whether the phenotype what you see in the EAE model is because of IL-12 or IL-23. So now this makes sense. So now here all the Th1 molecules, if you immunize, they are paralyzed. IL-12, there is a paralysis. P40, no paralysis. It could be because of IL-23, and IL-12 receptor beta 1, there is no paralysis, right? IL-12 receptor beta 2, there is a paralysis. So looks like it could be IL-23 playing a role in actually making the disease, making these mice resistant. So now basically this has been solved by a classical paper came out in 2003 by Dankoa, and what he has shown that if you have P35 knockout, P40 knockout or P19 knockout, so basically this is the IL-12, this is IL-23, and if you immunize them, P35 there is a paralysis, P40 they are resistant, and P19 there is a resistant. So looks like that it is IL-23 which is inducing, which is important for disease. IL-12 is not really important for this. In fact, IL-12 is making the disease worse. So if you don't have IL-23 in this setting, the mice are resistant to EAE. And this is how the discovery of IL-23 and its association with organ-specific autoimmune diseases came out. And later on, IL-23 and T cell connection came out to show that there is a Th1 and Th17 thing. So now when people isolated the T cells from these mice, so in P35 knockout mice, they are having paralysis, they're having more disease, and they have more Th17 cells, but no Th1 cells. In P40 knockout mice, no Th1 cells, no Th17 cells, because you don't have IL-12 and you don't have IL-23. They are resistant. And then if you have P19 knockout, there is a Th1 intact Th1 cells, but there is no Th17 cells. So it looks like that IL-23 together with Th17 is the key factor for inducing the paralysis in this settings. And at the same time, it was the time when IL-23 came out, and Judy Cho in Yale University has shown that the GWAS study, and it came out that IL-23 receptor and IL-23 P40 is important for the genetic association of inflammatory bowel disease. And this is a timeline how Th17 and IL-23 biologies are kind of opened up. Initially, it has been cloned in 1993, and later on at different time points, IL-17 receptor was cloned. In 2000, the 23 receptor and other subunits are cloned and characterized. And this is how the whole overall discovery happened in the field of Th17 and IL-23 biology. So now, I mean, it was not very clear how Th17 cells are differentiated, because IL-23 is definitely doing something to Th17 cells. But when people are analyzing the naive T cells, because the differentiation of T cells always begins with the naive T cells. So when people isolate these naive T cells from the mice and try to see whether they do express IL-23 receptor or not, and the answer was no, they don't express. So how IL-23 and Th17 connection was really there was not very clear, and whether IL-23 can induce the differentiation. If there is no receptor on naive T cells, IL-23 cannot really work, and that's what people have shown, that if you isolate the naive T cells, if you throw IL-23 onto it, they cannot become Th17 cells. So this is the time the paper came out from Cambridge, Brigitte Stockinger in 2006 in Immunity, where they're trying to understand how Th17 cells is differentiated. And what they have done, they cultured the 3 different cell types in a well. This is an in vitro experiment. They have taken the dendritic cells, Treg cells, and naive T cells. There is no Th17 differentiation. They collected the supernatant and do the IL-17 ELISA. The second setting, what they have done, they took the naive T cells, dendritic cells, Treg cells, and put LPS onto it. There's a differentiation of Th17 cells. So looks like LPS is doing something to this. And as we understand that LPS is actually primarily works on the dendritic cells, not on the Tregs, not on the naive T cells, because TLRs is primarily on the antigen presenting cells. So it looks like then it came out that if you replace LPS and dendritic cells with IL-6, and if you replace Treg cells with TGF-beta, now you have TGF-beta and IL-6, and these naive T cells can become Th17 cells. So this is how the differentiation factor has been identified in this immunity paper. And later on, the other paper came out from Vijay Kuchru's group, that if you just take the non-Treg cells, because at the time they have made the knock-in mice, so you can sort GFP positive, FOXP3 negative, and FOXP3 positive cells. And if you induce these FOXP3 positive cells with TGF-beta, they become Tregs. And if you induce FOXP3 positive cells, TGF-beta, in different cytokines, that's what the experiment they have done. And if you put the TGF-beta, you have nice induction of GFP, they become FOXP3, and IL-6 suppresses FOXP3 induction. So that's how, and if you collect the supernatant from this group, you see a lot of IL-17 in TGF-beta and IL-6 condition. And then they did the experiment. If you put TGF-beta alone on the non-FOXP3 positive cells, they become FOXP3. You can see nice induction of FOXP3 around 40%. And if you put TGF-beta plus IL-6, the FOXP3 induction is going down here from 40% to 10%. And you can see nice blimp of this IL-7 positive cells coming back. So it looks like that it's kind of reciprocal regulation of regulatory T cells and the IL-17 by TGF-beta and IL-6. And later on, it has been identified when you induce TGF-beta and IL-6, these Th17 cells, which is differentiating, produce IL-21. And we call them IL-21 as an amplification factor for Th17 cells, because it induces the amplification of Th17 cells. If you don't have IL-21 in this setting, the amplification is kind of stopped. And that's what it has been shown in this Nature paper in 2007 as a IL-21 is the amplification factor. So now what we can see is a differentiation factor, TGF-beta and IL-6. And during this differentiation, IL-21 is being produced, can work in the autocrine manner through IL-21 receptor and induces the amplification of Th17 cells. But still, we don't know what is the role of IL-23. So what we wanted to really understand at this point of time, making a kind of a knock-in mice, where we put GFP into IL-20 receptor. So every time IL-20 receptor is expressed, the GFP will be there. And when you put the GFP, it also kind of knock-in system. So basically, if you have the mice, which is a heterozygous, so one allele is wild type and one allele is GFP, you can consider this as a wild type mice, but at the same time, this is behave as a knock-in mice. You can use as a reporter mice. But if you breed them homozygous, then these alleles both are GFP, but they now cannot respond to IL-23. So this was a very nice system to understand the biology of IL-23. And then we did the experiment when you actually have IL-20 receptor knockout mice. These mice are completely resistant to EAE. They don't generate any disease. And if you take out the cells from these knockout mice, they don't have any Th17 cells. So based on this, we propose a role of IL-23, not only based on only one experiment, a lot of literature at that time. After amplification, IL-23 comes in and it stabilize the TH17 phenotype.
Could you please, Amit, time is very tight. Could you please pay attention to the time?
Just quickly, so basically, IL-17 is now is the first line of treatment for psoriasis. And briefly, I will just quickly 2 slides. Now, our lab is actually working on SARS-CoV-2, and what we have done here very quickly, the human ACE2 and SARS-CoV-2 RBD interaction, and you can see human, hamster, and the mouse. And the human and the hamster interaction of ACE2 and SARS-CoV-2 is so tight; it's very similar to human. Based on various interaction and thing, looks like hamster perhaps is really the right model, and a lot of literatures already came. And when we actually infect these hamster with the SARS-CoV-2 virus, you can see there's a nice viral load in the lung, and you can see there is a nice induction at day 2. There's a lot of lung pathology you can see. You can see a lot of splenomegaly at early days, and when you go into the later time point, around 14 days, when the virus resolves itself in the hamster, there's a splenomegaly is gone, and this viral titer is also going slowly back to normal. And we see a lot of lung pathology and different diseases score, lung injury. There is a kind of inflammation score. There's a mast cells as well as granulocyte score, what we have seen in our model. And one of the things which we have shown, which is very similar to what human settings are showing, that there's a kind of cytokine storm here, where you can see all these cytokines, irrespective of, whether it's a TH1, TNF-alpha, IL-13, IL-17. In fact, IL-9 is also there, are kind of upregulated at day 2. This is a day 2, this is day 4, day 7, and day 14 infection. And as days passes, the cytokine storm actually goes away, and these hamster actually gets better. So this is something which I wanted to share, and we have also found a lot of heart problem in this. We have done the complete lipidomics as well as metabolomics profile of these hamster, and it's kind of amazing data what we see, which is correlating cytokine storm and early heart disease problem in the SARS-CoV-2 in the animal model. Thank you very much, and I thanks Dr. Arora and the FIMSA team for giving me this opportunity for presenting this talk. Thank you.
Okay, thank you, Amit. As I don't think we have much time to answer the question raised by our audience. So anyway, we have to go very fast to answer some questions. So we have 7 questions. Is I can see. The first one is how to tell if a T cell subsize is a unique subsize or just a temporary shift in phenotype or functioning.
Yeah. So
could you please? Yeah.
This is so any T cells which is particularly TH1, TH2, TH17 cells, they have a master transcription factor, and this has been shown using the fate reporting fate mapping experiment. There is a certain degree of plasticity in certain things, but these are the real subset, and you can really see if you put either of this GATA3 T-bet or ROR gamma T in either of these cells, they become either TH1, TH2, and TH17, depending on the transcription factor you are actually infecting.
Okay, the second one is does pathophysiological conditions lead to L23 differentiation or secretion or release? So that means it's in which pathophysiological conditions the L23 released.
So IL-23 is primarily released like IL-12 from the antigen presenting cells, and certain like if you go back to the literature, the fungal pathogens, especially fungi, dactin pathway induces more IL-23. So it is like more antigen presenting cells is making more IL-23, and we don't know whether in the autoimmune setting how IL-23 is being induced. We have not yet any clue about that.
Okay, thank you. Another one is if we have the suppression of both interferon gamma and IL-4 in our setup, would it be correct to come to a conclusion that both the Th1 and the Th2 subsets response is getting decreased, or any further validation is needed?
Could you please just rephrase the question?
Yeah, this is a very long question. So I think the question means is, so if we have the suppression of the both interferon gamma and FO in our setup, would it be correct to come to a conclusion that both the Th1 and the Th2 subsize response is getting decreased? So I think maybe they want to ask about in which conditions interferon gamma producing Th1 and the L4 producing Th2 is both together getting decreased. So it is happen or not?
So in a physiological setting, it's really, see, in the physiology, it's never none or all phenomena, right? So only thing what you can see when neither Th1 or Th2 responses are there, it could be Th17 cells response will be there. And it has been shown in one of the paper in JEM where if you forcefully remove T-pad and get a 3 double knockout and then activate these T cells, they tend to, without even the differentiating cytokine of Th17, they tend to go to Th17 pathway.
Okay, thank you. So another one is, are there T-beta blockade or inhibitor available, and can they be used for treating hyperinflammatory or cytokine storm related immunopathologies as observed in COVID-19 patient?
I think as far as my knowledge, there is no small molecule inhibitors are available for Tibet, because I think if you really want to do neutralization, you can do interferon gamma neutralization. Those antibodies are available, but not sure, it's not there. There are a lot of
another one.
a lot of inhibitors for ROR gamma T. There are a lot of inhibitors for ROR gamma T.
Yeah, I totally understand. Even it is a very good question. So another one, what is the role of the interleukin 17 and FOXP3 T regulatory cells in women with recurrent pregnancy loss? This is a disease in women, so means recurrent pregnancy loss. What is the role of the Th17?
So normally whenever you have a pregnancy, pregnancy is kind of tolerance, setting of tolerance. So basically you see a lot of upregulation of FOXP3 cells as well as NKT cells. I don't know whether IL-17 has been tested, but I have no idea about this, whether recurrence loss of pregnancy, you have disbalance of Th17 and Treg cells.
Okay, so the last one is interleukin 12A expression is not correlated with interleukin 12B. So if I want to study the expression of L12 pulse, which subunit is more reflect the expression?
Yeah, so basically, if you want to do, you have to do is IL-35 for sure, and but if you need to do IL-12 bioactive ELISA, which is P70. So P70, you need to do ELISA for P70 in your settings, because IL-35 can also homodimerize. So doing real-time PCR for IL-35 may not be a good answer, but you need to do bioactive ELISA for IL-12.
Okay. Thank you very much, Amit. So I learned a lot. So you give the very wonderful talk on a comprehensive review of the T cell and its cytokines and differentiation. Thank you very much.
Thank you.
So we move to the next speakers. Is Professor Hong Yan Wang. She is the professor of Shanghai Institute of Biochemistry and Cell Biology, Chinese Academy of Sciences. She's working to identify key kinases, enzymes, and signaling molecules in macrophage and T cells that mediate cell migration, inflammation, and immune surveillance. Especially, she is interested in how lipid regulate function of macrophage, CD8 positive T cells, and MDSC during antiviral and anti-tumor response. as a corresponding author. She has published more than 20 papers, including those published in Immunity, Nature Microbiology, Journal of Clinical Investigation, Journal of Experimental Medicine, and so on. Today, her lecture title is The Function and Mechanisms of Macrophage Against Infections. Hong Yan, please.
Okay. Hi. Can you hear me properly?
Yes, again. It's very good.
Yes. Thank Professor Tian's kind invitation, and I enjoyed this fantastic meeting very much. So I thank the meeting organizer to provide me this wonderful chance to talk about my understanding about macrophages. So now I will share my PPT and start my lecture. So it may take, okay. So today I will talk about the function and mechanism of macrophage against infection. As we have learned in the past lectures, macrophages are one of the key cell types in innate immunity. Very similar to dendritic cells, macrophage can also act as a bridge to link innate immunity and adaptive immunity. There are several features of macrophages. For example, macrophages are typical or professional antigen presenting cells, which can present antigen to T cells and activate T cells. And also there are several different characters. For example, macrophages can secrete many different kinds of cytokines during infection or even during anti-tumor immunity. So let's start to know more about macrophage. A macrophage can divide from monocyte. And in these pictures, we know that in the bone marrow, HSC can be differentiated into myeloid progenitor cells. And these progenitor cells can further differentiate into monocyte. And the monocyte in the bloodstream can migrate into different tissue and become macrophage. And recent publications suggest that apart from bone marrow, macrophages can located in different tissue or organs and derived from different sources. For example, the microglia in brain can derive from Yoxide, and the Kupffer cells in liver can derive from Yoxide as well as HSC. And macrophages can also located in lung, spleen, skin, heart, kidney, peritoneal, and intestine. So all these organs are very important for the host. And since macrophages can mediate inflammation, so macrophages can take part in inflammation-associated diseases in all these kind of organs. So that indicates macrophages are very important. And I think there are several features about macrophages. First, macrophages can express multiple receptors. These receptors can help macrophages to sense different stimuli in the microenvironment. For example, it can express Fc receptor, complement receptor, which help macrophages to sense bacteria or other pathogens via the help of complement C3b or the help of the Fc fragment of antibody. And also macrophages can express MHC, which can present antigen to T cells. And macrophage can also express CD40, CD8 T, CD86. These are ligands for T cell co-stimulators. And in certain microenvironment, for example, in tumors, macrophage can also express ligand for PD-1, for instance, PD-L1 or PD-L2. So these molecules can determine whether macrophage can activate T cell or cause T cell exhaustion. Except for these except for these receptors, as we have learned a wonderful talk from yesterday's speaker, Professor Taylor. He talked about pattern recognition receptors, PRR. Macrophages can definitely express PRR, which can allow macrophages to sense pathogen-associated molecular patterns, PAMs, so that they can produce different kinds of cytokines. For example, upon infection, if the host produces antibodies, the FAB fragment combining to different antigens, while the conserved FC fragment of this antibody can be recognized by HC receptor on the macrophage. In this cartoon, the bacteria is indicated as a red circle. It can be covered by antibodies, and the FC fragment of the antibody can bind to the FC receptor on macrophages, which can initiate phagocytosis. And the phagosome can then fuse with the lysosome to form this phagolysosome. And inside the phagolysosome, pathogens, especially bacteria, can be digested and killed. And we also learned how complement is very important for our host from yesterday's talk by Professor Sahu. And here, if the host produces complement during infection, the complement such as C3b combines to bacteria and drive bacteria towards to macrophage. And via the binding to the complement receptor CR1, then it can initiate endocytosis process or form phagocytosis. And this process can also kill the invading pathogens. And if we, as the host, we have very strong immune response that our body produces antibody together with the complements, then these two effectors can work together to greatly enhance the binding to the pathogens and also enhance the ability for phagocytosis, so that in this cartoon, it's simplified to understand that the response can be further enhanced as 4+. So except for macrophages, other phagocytes, for example, neutrophils, can also use FC receptor and the complement receptor to mediate phagocytosis. And this is very important for the innate immune cells against infection. And the second feature about macrophages is that they can produce over hundreds different kinds of biological active substance. One of the most important substances are cytokines. These cytokines can be inflammatory cytokines, such as IL-1, IL-6, TNF-alpha, and type 1 interferon. And they can also be anti-inflammatory cytokines, IL-10, TGF-beta, that we will talk about later. And except from these typical cytokines, macrophages can also produce growth factors such as FGF, VEGF, or chemokines such as CCL2, CCL5, etc., which can recruit many other different kinds of innate or adaptive immune cells to the site of infection, and then they can work together to clear the pathogens. And also macrophages can produce many different enzymes, such as proteinase, lysosome. These enzymes can help macrophages to clear the invading pathogens. And macrophages can also produce complements, blood coating factors, and the PG or LT. This substance can help macrophage to manipulate the scale of inflammation so that macrophage can be involved in tissue repair, anti-infection, or anti-tumor responses. And because macrophages can express multiple receptors, can release different substrates, and the third character of macrophages is that they can be activated and different microenvironment. So that means their function is plasticity. So they can switch, they can be activated, for example, by bacterial LPS or cytokine interferon gamma, and the resting macrophage will be polarized towards M1 macrophages. In M1 macrophages, the transcription factors NF-kappa B, STAT1, IRF5 are activated. And then it will enable M1 macrophages to produce large amount of pro-inflammatory cytokines, including IL-1, IL-12, IL-6, TNF-alpha, chemokine, etc. And also M1 macrophage can produce large amount of ROS, NO, and enzymes to facilitate the killing of different pathogens, including bacteria or fungi. But if macrophages is stimulated by other cytokines, typically by IL-4 or IL-13, they can differentiate into M2 macrophages. In M2 macrophages, some transcription factors, including PPAR gamma, IF4, STAT6, are activated, so that M2 macrophages can produce their marker, including arginase 1, YM1, FITS1. And M2 macrophage also produce anti-inflammatory cytokines, IL-10. When M2 macrophage engulf apoptotic cells or tumor cells, they also produce large amount of TGA-beta. So if this kind of macrophages is located in injured tissue or tumor, they produce large amount of IL-10 TGA-beta to either facilitate repair of the tissue or facilitate the growth of the tumor. For example, in tumor, this kind of macrophage is named as tumor-associated macrophages, in short, TAM. And TAM can be induced by MCSF, IL-4, IL-10, TGA-beta. And once TAM is formed, they produce again higher amount of IL-10 TGA-beta, but the lower expression of MHC. So their professional APC ability is reduced. And also these anti-inflammatory cytokines could inhibit Th1 differentiation, but promote Treg differentiation. Therefore, TAM can immune suppress T cell function. And also TAM can produce several different growth factors, such as VEGF, EGF, FGF. And these growth factors can promote angiogenesis, or it can act directly on tumor cells to promote tumor growth and tumor invasion. So except the above information that I mentioned, my topic is focused on pattern recognition receptors as well. And again, as I said, since Professor Taylor has given a very introductive, informative lecture yesterday, I will briefly introduce PRR. So PRR are pattern recognition receptor, which can recognize pathogen-associated molecular pattern, i.e. PAM, of different pathogens, including bacterial, DNA virus, RNA virus, or fungi. And the pattern recognition receptors can also recognize self-molecules released from necrotic cells or damaged tissue, including DAMs. So once these pattern recognition receptors are activated, it can allow macrophage to produce different cytokines. And Toll-like receptor is one of the PRR. They are transmembrane receptors and detect extracellular or endosome microbial. So after Toll-like receptor signaling, macrophage can produce cytokines as well as type 1 interferons. And the RIG-I-like receptors is located inside the plasmid. It can recognize RNA virus and generate RIG-I marvelous pathway to clear RNA virus. And the CGAS sting can sense cytoplasmic DNA, including DNA virus or the damaged or the DNA from tumor cells. So the CGAS sting pathway can initiate the production of cytokines as well as type 1 interferon. And NOT-like receptor, NLR, also belongs to PRR. They located in cytoplasmic. They can detect intracellular bacteria to activate inflammasome so that IL-1 beta and IL-18 can be matured and released by macrophages. And the C-lectin receptors also belongs to PRR. They can detect actual microbials. So based on these knowledges, we now understanding why macrophages can recognize bacteria and destroy bacteria. Because they have these pattern recognition receptors, or they have Fc gamma receptor or complement receptor. And after recognizing the pathogens, many different signaling pathways can be activated, and they eventually activate different transcription factors in the nucleus, including NF-kappa B for the production of inflammatory cytokines, or the transcription factor IF3 for the type 1 interferon production. We are particularly interested in Toll-like receptor signaling. So in short, these signaling pathways are mainly divided into mediated dependent pathway or mediated independent pathway. In the left side, the mediated dependent pathway is labeled with yellow color. So downstream of mediated, the adapters IRAC, TREP6 can be linked to either TAC1 IKK complex to activate the transcription factor NF-kappa B, or TAK1 can link to the MAP kinase, including ERK, JNK, P38, which can activate the transcription factor AP1. And this mediated dependent pathway can generate different kinds of pro-inflammatory cytokines and chemokines. And in the right side, it is mediated independent pathway, which is also called the TREF dependent pathway. In this pathway, downstream of TREF, it involves TREF3, TBK1, IKK epsilon, and this can activate IRF3 for the induction of type 1 interferon. And this paper published in Science China by Professor Chao's group, it elucidated a mediated dependent TLR pathway or TREF dependent TLR pathway. Here I wouldn't spend time to go further. So I will take this chance to introduce some of our projects related to Toll-like receptor signaling. Although Toll-like receptor signaling is critical for macrophage to remove pathogens, However, excessive inflammation also causes certain types of diseases. For example, the acute cytokine storm is one of the key reasons leading to sepsis or septic shock. And also, toll-like receptor signaling can generate chronic inflammation, and this will contribute to the development of certain types of cancers, including liver cancers, colon cancers, and stomach cancers. So can we identify any new kinase or enzymes to prevent excessive inflammation, and so that we can prevent sepsis or block HCC development? Here we show one of our work. We use bacteria to infect macrophages and screen the expression levels of surface receptors. Some receptors, including IL-6 receptors or chemokine receptors, will be reduced upon bacterial infection. In contrast, we found that upon bacterial infection, macrophages can significantly upregulate VEGFR3 expression. VEGFR3 is vascular endothelial growth factor receptor 3. From its name, you know that previous study found that this receptor is mainly expressed in endothelial cells to promote endothelial cell proliferation and thus contributed to lymphoangiogenesis. However, once VEGFR3 is mutant in patient, this patient can also suffer from reoccurred infections that indicate the upregulated VEGFR3 on macrophages may play a role in innate immunity. And in this study, we also find the ligand VEGF-C is also upregulated in sepsis patient. And both VEGFR3 and the ligand VEGF-C can be inducibly expressed via TOL4 NF-κB pathway. And once VEGF-3 bind to this receptor, they can activate PR3 kinase AKT pathway to promote toll-like receptor 4 endocytosis, therefore to suppress NF-κB activation. And they also promote IL-10 production. So in short, VEGFR3 is induced by toll-like receptor 4 signaling, which can then inhibit toll-like receptor 4 pathway, so they can act as a brake to balance the inflammation and avoid sepsis. So is that a possible clinical application if we target VEGFR3? Later, several different groups use different models and demonstrated the key role of VEGFR3. For example, this paper published in GCI, they using IBD patient sample or IBD mouse model and found that VEGF-C is enhanced, and VEGF-C can activate VEGFR3 on macrophages to inhibit infection. So that later a review published in Nature Review Rheumatology cite our work and think if we can activate VEGFR3 signaling, we can inhibit inflammation, and also we can promote lymphopenia function, so that it has two different roles for the therapeutic invention for RA patient. And later, there is another work published in Cell Reports and found that macrophages in a tumor microenvironment can express VEGFR3 at high level and secrete VEGF3 to facilitate the tumor cells against the chemotherapy drugs. So if we block VEGFR3, it might enhance the possibility to treat tumor, to prevent tumor metastasis. And the toll-like receptor signaling can also promote liver cancer development. And in others report, if you mutant toll-like receptor 4 in hepatocyte, even if the mice were induced by DN plus CCL4, these TLR4 mutant mice were protected from HCC. And if we use wild-type mice and put them into germ-free facility, they cannot be initiated or recognize bacteria, then there is no chronic inflammation, and this germ-free mice also protected from HCC. So we generate inflammation-associated HCC model, and we can find that in the liver tumor, there are a lot of macrophages. We isolate these macrophages, and we found that one of the tumor suppressor, STK4, not only downregulate their expression in tumor cells, but also downregulate the expression in tumor-associated macrophages. And if STK4 is knocked out or downregulated, macrophage can produce a much higher level of IL-6 at early stage. And also these mice can form more liver tumors at late stage. But as a tumor suppressor, we cannot easily upregulate STK4 expression or kinase activity. And in this work, we finally figure out that IRAC is further downstream effect. If STK4 is reduced their expression level, IRAC is stabilized at protein level, so that we can treat these mice with IRAC inhibitor to block IL-6 production, and later it can also greatly reduce liver tumor formation. That makes us to wonder if we can identify a portion of patients which reduce STK4 expression, and then maybe at the early stage, we can use IRAC inhibitor as a drug to block inflammation and block HCC development. And also, if we look at the toll-like receptor signaling, we notice that ubiquination play a very important role to manipulate the inflammation response. Can we identify new E3 ligase? So we use this siRNA screening system. In this multi-well plate, in each well, we use 4 siRNA to target one E3 ligase. This can allow us to effectively knock down any E3 ligase. So after that, this peritoneal macrophages was stimulated with LPS for 6 hours, and then we harvest the supernatant to measure IL-6 production. This screening system is reliable and repeatable. We have identified HRD1 as a hotspot. HRD1 is located in the ER. It can promote degradation of misfolded proteins in the ER to reduce ER stress and prevent cell apoptosis. That's the HRD1 classical function. It's named ERAD. And because of this ERAD, so that synovial cells upregulate HRD1 expression, and this can allow the synovial cell proliferate and cause RA. And our model found that HRD1 does not affect macrophage apoptosis upon infected with different bacteria, but it can induce inflammation via I-kappa B alpha degradation. I-kappa B alpha is an inhibitor for the inflammation. It can bind to NF-kappa B and keep NF-kappa B in the cytoplasm. But once I-kappa B alpha is degraded, NF-kappa B can enter into the nucleus to mediate inflammation. So in wild-type cells, if the E3 is present, I-kappa B alpha is degraded it's degraded, and P65 entry to the nucleus to promote inflammation. If we knock out this E3 ligase, we can reduce the amount of P65 in the nucleus. We can inhibit inflammation. So later we propose this model that we have identified an E3 ligase called HRD1, which can cooperate with the surface toll-like receptor 4 to promote inflammation. If we target HRD1, we can protect the mice from septic shock. And a nice paper from another group, Professor Fang Deyu's lab in Northwest America, they knock out HRD1 in liver cells and find that these mice are protected from high-fat induced liver damage. And HRD1 is closely related to glycerol synthesis. So we wonder whether glycerol metabolism plays any role in innate immune response. Glycerol are key component in the cell membrane and the vesicle. And during viral entry, if we reduce glycerol levels in the plasma membrane, we can inhibit HIV entry. And also upon pathogen infection, macrophages can change metabolic enzyme expression levels, and the related metabolic product can affect macrophage ability for produce inflammation or type 1 interferon. And this cartoon illustrates how glycerol metabolism is regulated by different key enzymes. These enzymes are labeled with yellow, and the three type of glycerol products have been recently indicated to play important role against the viral infections. So let me introduce one of the product. If we knock out glycerol synthesize enzyme SREBP2, this will generate much more type 1 interferon by macrophages. And the type 1 interferon can bind into the receptor and induce interferon stimulated genes. And this ISG, for example, one of the glycerol metabolism enzyme, CH25H, is induced by type 1 interferon. And this CH25H can further convert glycerol into 25-hydroglycerol, 25HC. And this nature product, 25HC, can prevent HBV or VEC viral entry or replication. If we look at glycerol metabolic enzymes, there are a lot of different other unknown function enzymes in the innate immunity. So can we identify any new glycerol enzyme or products in antiviral infection? So we use the three different samples to screen differentially expressed metabolic enzymes. And as shown here, we found that upon RNA virus, DNA virus, bacteria, or type 1 interferon stimulation, the enzyme DHCR7 is reduced their expression level. DHCR7 is 7-dehydroglycerol reductase. In this map, DHCR7 can convert 7-DHC into glycerol. So if DHCR7 expression is lower, what will happen? We knocked out DHCR7 in macrophages and then infected the macrophage with DNA virus, RNA virus, or poly-IZ to activate toll-like receptor 3 pathway. And in all this kind of treatment, if we knock down DHCR7, it can produce much more interferon beta. And this is related specifically to the activation of IF3. The TPK1 phosphorylation is not altered. Since it's an enzyme, we can find an inhibitor, AY9944, to block the activity of DHCR7. And this inhibitor can also promote macrophage to produce much more interferon upon viral infection. And it also promoted activation of IF3. And importantly, if we use this inhibitor to treat mice, it also protects the mice against the VSV infection or H1N1 infection. And upon inhibitor DHCR7 activity, it can cause the upstream product 7-DHC accumulation. So what's the role of this nature product? We add 7-DHC to the cell culture medium. It can promote macrophages to clear VSV and to activate IF3. And because of the time, I think I can summarize the mechanism here. We think that we have identified a key enzyme called DHCR7. If we use inhibitor to block its activity, it can cause the accumulation of 7-DHC. And 7-DHC can activate PR3 kinase and the downstream AKT3. And interestingly, AKT3 is barely or very expressed at a very low level in resting macrophages. Upon infection, AKT3 can upregulate expression level and can be activated by 7-DHC treatment, which can then bind to IF3 and phosphorylate the serine 385 site in the IF3 to activate IF3. Therefore, macrophages can produce much higher level of interferon beta against the DNA virus, RNA virus. So we propose that DHCR7 might be a very good drug targetable candidate. And all this detailed data I will skip. And finally, I want to thank all my team members and thank my grants from mainland of China. And also thanks for your attention. If you have any questions, I'm happy to answer. Thank you very much.
Okay, thank you for the wonderful talk and very comprehensive overview on macrophage and also molecular mechanisms to control the function of the macrophage, and people should be learn a lot. So there are several questions to ask you, and just depending on the time left. The first question is, what is the role of the CSF1 receptor or CFM kinase in deciding macrophage polarization and plasticity? So this is a very general question. You can give some comments on that.
Yes, it's very nice question and very important. I think recently there are several papers published, such as in Nature Immunology, about how MCSF can promote M2 macrophage polarization. So if MCSF can promote M2 macrophage polarization, it will via the receptor called CSF1R. So that has been demonstrated by a couple of articles.
Okay, thank you. So a second question is how much the role played by macrophage in autoimmune disease, in particular SLE?
Mm-hmm. Yes, that's very good question as well. In the pathological of SLE, so it's mainly triggered by self-DNA, and it causes the autoantibody production, mainly by B cells. But macrophages are professional antigen presenting cells, which can activate T cells, and it can also activate B cells, because T cells maturation, proliferation need cytokines IL-4, and IL-4 can be produced by Th2 cells. So macrophages can facilitate SLE via secreting this kind of cytokines or via facilitated Th2 development. And also, if we learned about pattern PRR, we know that macrophages have toll-like receptor 9, which can recognize self-DNA to generate a pathway and then produce high level of interferon. And interferon can also contribute to the development of SLE. So there are many different mechanisms that can be controlled by macrophages during the development of SLE.
Okay, thank you. Another question is, and the audience is very interested in SREB2 inhibition to promote interferon production and their role in antiviral immunity. So their question is, they want to know what about the anti-tumor? So it is works or not in the same way.
Right. I don't know whether people target SREBP2 directly on tumor cells, but if we target them in macrophages, it can promote type 1 interferon production. And once type 1 interferon is produced, it can promote MHC or T cell function, so that it can contribute to anti-tumor responses. And definitely interferon or interferon gamma can promote a type 1 response. SREBP2 is a key enzyme to synthesize glycerol. If we block glycerol in tumor cells, it might drive tumor cell apoptosis. So whether we can target the SREBP2 to enhance type 1 interferon in innate cells and also target SREBP2 in tumor cells to promote tumor death. So whether this different mechanism existed, I think this needs further elucidation.
Okay, so another question is any of the information about the VEGF receptor 3 in a clinic or preclinical practice in a disease? I mean, the inhibitor or blocker of the VEGFR3 in a preclinical or clinic studies. What is about information? Can you give some of the information about that, or if this target is still useful for the people?
Right. In preclinical trials, I know that some companies have targeted VEGFR3 and they are producing inhibitors, and they want to treat certain tumors to block tumor metastasis. And in this family, there is another two members called the VEGFR1, VEGFR2, and they have the common ligand called VEGF-A. And VEGF-A monoclonal antibody or monoclonal antibody against VEGFR1 or VEGFR2 has been extensively used currently in the preclinical treatment against the tumor. So there are, I think there are potential space to develop VEGFR3 and VEGF-C as the drug candidate as well.
Okay, the last question is any role of the pH of tumor microenvironment in regulating the production of VEGFR3 by macrophage?
That's a very interesting question. I haven't
yeah, pH of the tumor microenvironment
Very
in the production of VEGF receptor 3 by macrophage.
Yeah, we know that in tumor microenvironment, pH is lower, so that can maybe facilitate other key effector expression, such as HIF-1 alpha is also involved. And in our lab, also test whether lower pH can promote any enzyme expression, especially in the regulation of glycerol metabolism currently. And we haven't tested directly whether lower pH can promote VEGFR3 expression or the ligand VEGF-T expression in macrophages. But if you are interested, I think it's very nice topic to have a try.
Okay, thank you for both of the speakers and the question answers, and also all the participants. Thank you, thank you,
Thank you.
thank you for your attention, and also hope you have a good lunch. Thank you.
Thank you.
Okay, I have to close the session.
Thank you, Dr. Tian, for the wonderful moderation and the speakers for taking care of the session so well. So may I request Dr. Arora to make any announcements, if any?
All right, thank you, Dr. Tian. Thank you,
Yeah.
Hongyan. Thank you, Amit Awasthi, for a very nice this session. We are going to have lunch break, and then the post-lunch session, we plan to start 10 minutes earlier, around 2:30 now. So this is because one of the speakers has to go somewhere, so he wants to start early. So we'll start at 2:30 instead of 2:40. So all are requested to come back early and join at 2:30 now. Thank you.
Okay,
Have a
thank
good
you. Thank you very much. Thank you.